Hi, welcome everyone on behalf of the National Academy of Sciences and Engineering and Medicine. We want to welcome you to our workshop today. It's a two-day workshop on the interactions of biotechnology with human physiology and function. I am Kavita Berger, the Director of the Board on Life Sciences, and we are really pleased as part of our Standing Committee on Biotechnology and National Security to be hosting this workshop with you. I want to introduce our Vice Chair of the Standing Committee to also welcome you today.
Hi, good morning, everyone. I'm Diane Deulis from National Defense University. I want to say welcome to everyone as the co-chair of the ad hoc committee and the co-chair of the standing committee. Thanks to everyone for coming and attending, and all those who are attending virtually. I want to thank the other co-chair, Dr. Todd Coleman of Stanford. And also I want to thank Elliot Chekhov, who's going to be leading us through the next 2 days, which I hope is going to be very exciting. Human physiology and function, there's a lot of chatter and talk about human performance and human physiology and all the things that we can do with it. And I think this topic that we're going to tackle over the next 2 days is actually a space that's ready for some real innovation and exciting things to happen. So I'm looking forward to all of the talks we're going to hear. So with that, I will turn it over to Dr. Chekhov.
Oh, is this somebody's? Well, welcome. I'd like to welcome everyone who's here in person and all those who are with us virtually. My name's Elliot Chekhov. I'm a professor at Harvard Medical School and chair of the Department of Surgery at Beth Israel Deaconess Medical Center. If I could have the first slide, please. Just one minute. I don't know if no, it's not mine. It's for my introductory presentation. I think you had it correct. Perfect. All right. Well, again, I'd like to welcome everyone to this two-day workshop on the interactions of biotechnology with human physiology and function. I'd like to thank all of the participants, those who are here in person, as well as those who are speaking virtually over the next 2 days. Just need, if you can make sure that the clicker is working. Nothing in biology makes sense except in the light of evolution. That was a principle that was put forward by Theodosius Dobzhansky, a geneticist and evolutionary biologist who worked in the mid-portion of the 20th century. Evolution occurs through a process of acquiring adaptive features that allow us to survive and reproduce better than our competitors, a process of natural selection. Over hundreds of thousands or even millions of years, we have intimately co-evolved with the microbial world that surrounds us. While adaptive features are typically considered inherited human traits that ensure relative reproductive success, that view may be overly narrow. Our human genome and our microbiome, which we have coexist, both work in concert with intertwined goals in mind. Not to live long, healthy, and happy lives, but to survive and reproduce. Health and wellness are merely byproducts of that goal. The challenge we face in the 21st century is a growing evolutionary mismatch. Our body evolved and adapted over time to succeed in one environment, and for a variety of circumstances, we now find ourselves in another. Evolution is not simply about biological evolution, a series of inherited genetic variations that change over time. Evolution is profoundly influenced by cultural evolution. The agricultural revolution, a transition from hunter-gatherers to farming, and subsequently the Industrial Revolution, have been cultural transformations that radically altered our environment. Cultural evolution is currently the most profound force for change for our entire planet. How we continue to alter our environment for good and for bad is continuing to play out in the food we eat, the medications we use to fight disease, how we work, and how we sleep. Some of these interactions are beneficial, but in other instances, our bodies are poorly or inadequately adapted to the novel environment that we have created through culture. Technology has altered and continues to alter human physiology and microbial physiology for good and for bad. A case in point is the topic of our two-day workshop, the myriad of interactions between our gut, our brain, the food we eat, and the microbial world in which we coexist that serve to ensure our survival and our reproductive fitness. Health, wellness, long life, and happiness can be byproducts of these interactions, but conditions of maladaptation association with a genetic predisposition can lead to severe illness and poor health. In coevolution with our microbial environment, we are increasingly aware that microbes are critical to our ability to effectively respond to dangerous pathogens and our capacity for normal human growth and development, tissue repair, and homeostasis. To effectively monitor, function, and survive within our environment, we also have a growing appreciation that our neurological and immune systems have co-evolved with our microbiota. Despite the original concept that only our genes directly influence health and disease, a largely overlooked source of genetic variations stem from the microbes living on and within us. Indeed, we know now that these microbes, which we have inherited from one generation to the next, can influence every aspect of host physiology, including brain function. Thus, in a holistic view, we should see the intricate relationships between the hosts and its microbiota as a single powerful biological unit that promote health and well-being. Our microbiota, and in particular those communities within the gut, have been incorporated into the sensory systems that we use to survey our environment, and importantly, provide information on the physiological state of our body. Our collective microbes and their genes regulate the connections between the gut and brain to optimize our capacity to sense and respond to our environment. Moreover, as we will hear today, recent studies show that one can even leverage gut microbes to treat specific symptoms of brain disorders, a concept that was inconceivable a few years ago. In this dialogue of host and microbial genes also talk to the immune system. The gut immune system is imprinted by our microbiota as soon as we are born to promote tissue repair, to ensure our ability to rapidly respond to pathogens, and in the process, influences our neurological development, including social behavior and memory. Importantly, growing evidence supports that our nervous system, in turn, serves as a rheostat for the gut immune system. Moreover, immune cells in the gut and the brain constantly survey environmental factors that are central components to the gut-brain axis, eliciting responses that inform upon our physiological state. Indeed, new bioelectronic advances now provide a means to stimulate specific nerves to limit maladaptive responses responsible for a variety of inflammatory diseases. It is increasingly clear that a wide variety of metabolic, immune, and neuronal pathways mediate communication across the microbiota, gut-brain axis, as a homeostatic system for health and wellness within an ever-changing external environment. Defining the specific signals responsible for communication along these pathways may help us better understand those circumstances in which a mismatch with our environment has led to disease, along with opportunities to intervene. Understanding the circuits by which our nervous system interfaces with the gut immune system may provide us with a means to modulate neuronal signals, to dampen maladaptive inflammatory responses, or to tune our immune system to prevent or treat neurobehavioral and neurodegenerative diseases. Dissecting the contribution of host and microbial genes to specific behaviors or host phenotypes will not only provide a better understanding of the mechanisms underlying these processes, but also could lead to new therapeutics. A primary function of the gut is to ensure adequate nutrition critical to human health, wellness, and survival. The nutritional value of food is influenced by the gut microbial community, but what we eat can drive changes in gut microbiota, including microbial diversity, gene expression, and metabolic pathways. Modern diseases are on the rise that are inherently disorders of diet and dysbiosis. We are what we eat. If we define the contribution of the microbiome to specific behaviors or host conditions, we should be able to develop microbiota-directed therapeutic foods to treat a variety of conditions. That said, the opportunity presented by this workshop on the interactions of biotechnology with human physiology is not simply the potential to discover new drugs or develop new devices, but the promise to achieve a much deeper understanding of the circuits and mechanisms between our gut, brain, and our microbiota, and its contribution to health. In the words of Daniel Lieberman, a professor of evolutionary biology at Harvard, to help us break out of the pernicious feedback loop of disevolution between culture and biology. A longstanding challenge. Get the next slide, please. A longstanding challenge in the advancement of fields is an ever-increasing requirement for specialization, along with a need for consilience, or jumping together of knowledge by linking facts and fact-based theories across disciplines to create a common groundwork of explanation. A unique characteristic of those individuals who broadly conceived of the notion of the ecosystems in which we coexist, Alexander von Humboldt, Charles Darwin, and perhaps most recently E.O. Wilson, was their ability to draw on highly varied, disparate, complex bodies of knowledge to solve problems. The capacity for synthesis led Humboldt in 1834 at the age of 65 to begin writing a multi-volume treatise, Cosmos, to unify diverse branches of scientific knowledge and culture, a book on nature, which sought to provide a holistic perception of the universe as a single interacting entity. In the same year that Humboldt was embarking on an effort to bring all fields of science together, William Whewell, a Cambridge University historian, was the first to introduce the term scientist, heralding the beginning of the professionalization of the sciences and the hardening lines between different scientific disciplines. 200 years ago, science moved away from nature into laboratories and universities, separating itself into distinct disciplines, encouraged by the production lines of the Industrial Revolution. Whewell recognized both an increasing proclivity of separation and dismemberment in the sciences and the need for consilience of inductions. Reality is not approached by a single academic discipline, but by a consilience between them, a combining of knowledge that may be derived from the fields of ethics, the social sciences, biology, and the physical sciences. The challenges of simultaneously achieving breadth, depth, and integration of knowledge across highly specialized technical and disparate fields is no mean feat. In an age of increasing specialization inherent to an accelerated rise of new knowledge, it would be a matter of hubris not to acknowledge the increasing difficulty for any one individual to master multiple disciplines. But now more than ever, there is a need to mind the gaps and draw attention to knowledge that may otherwise disappear into the spaces between disciplines as they are currently defined and organized. There is a need for consilience, a consilience of reduction through the dissection of a phenomena into its component parts, and a consilience by synthesis with the prediction of higher order phenomena from more basic principles. How will we surmount these barriers? create a balanced workforce of specialists and generalists, ensure that governments, universities, foundations, and industry nurture a habitat that fosters new mechanisms of innovation, advances in technology, and solves problems. This will be a frame for our workshop that examines opportunities and challenges in leveraging our growing understanding of the gut, microbiota, immune neurological access. I'd like to introduce the first speaker for this morning's session, Dr. Sriram Chandra Sakharan from the University of Michigan. Dr. Chandra Sakharan is an assistant professor of biomedical engineering and leads a systems biology and drug discovery research program. He obtained a PhD in biophysics and computational biology from the University of Illinois, Urbana-Champaign, and pursued postdoctoral training at Harvard. Dr. Chandra Sakharan develops computational tools that, together with approaches in machine learning and AI, simulate thousands of metabolic reactions in human and microbial cells. This work provides a path towards engineering microbial metabolism for manufacturing new drugs, manipulating stem cell epigenomes, and understanding metabolic disorders. His work in identifying new drugs and drug combinations is directed at slowing the evolution of antibiotic resistance among microorganisms. Last year, Dr. Chandra Sakharan was recognized by MIT Technology Review as one of 35 innovators under the age of 35. Dr. Chandra Sakharan, I will turn the podium over to you. Thank you.
Thank you for the kind introduction. I'm really excited to be part of this workshop. And I should also say that I'm under some pressure because my mother-in-law is watching this live on the webcast. So hopefully I do well. So my lab develops computational tools for precision health. And today I'm going to talk about a key biological mechanism that links gut-brain interactions, that is through the metabolic epigenetic axis. That helps understand how changes in diet or environmental metabolites can influence neurological behavior and other metabolic disorders. So what is this metabolic epigenetic axis? So cellular metabolism involves thousands of chemical reactions that break down nutrients in the diet and provides precursors for synthesizing DNA and other key biomolecules. And another new discovery that was recently made is that many of these metabolites can directly influence gene expression of cells by chemically modifying histone proteins or the epigenome. So for those who don't know what epigenome is, so epigenome has multiple components. The key component is what's called histone proteins that wrap around DNA in cells. And chemical modifications of these histone proteins can release the DNA, resulting in turning on of genes, or it can make it more tightly bound to DNA, resulting in gene repression. And the important thing is that these histone proteins are highly sensitive to the metabolic state of the cell. So any changes in metabolism can result in modification of histones, resulting in wide changes in gene expression of a system. And it plays a key role, especially in this gut-brain axis. But there was one really cool recent study where they found how alcohol gets metabolized into acetate in the liver, and this acetate then goes through the blood to the brain and modifies histones in the hippocampus. So that gives you a connection between like dietary metabolism to neurological behavior, and it happens at the molecular level through this metabolic epigenetic axis. This is one example of many others that people have found through the microbiome and others, where metabolites produced through the microbiome or others can modify histones, resulting in changes in gene expression and cellular state. And so I'm going to talk about this key interaction at this metabolic and epigenetic level, how it can influence various physiological processes. So I showed a very simple example how acetate can modify histone acylation, but there's actually a pretty complex crosstalk between metabolism and epigenome. So for example, just to give two key epigenetic marks, histone acylation depends on acetyl-CoA, a key metabolic precursor that is involved in numerous metabolic pathways like fatty acid metabolism, TCA cycle, and the levels of acetyl-CoA can influence the acetylation of histones. And the removal of acetylation called deacetylation depends on NAD, which is a key redox precursor involved in hundreds of metabolic reactions. And similarly, methylation depends on numerous metabolites, like folates, methionine. All this can influence methylation. Similarly, demethylation relies on like iron, TCA cycle metabolites too. So there's a really complex crosstalk between, metabolism and epigenome. So it's really hard to predict what happens when there's a change in diet or metabolic environment on how that could impact the epigenome of the system. And so if you want to understand, like at a big picture level, these high level correlations that people have found on, diet and behavior, we need to figure out at the molecular level how changes in these environmental compounds or metabolites influence these metabolic pathways in a cell, how that influences the epigenome, which then results in, pathogenic states or different changes in behavior. So my lab's been developing computational models to tackle this complexity. We've been using a combination of traditional engineering methods that can simulate thousands of metabolic or biochemical reactions in a cell. And we're combining that with cutting-edge machine learning tools. The reason we are doing that is that while traditional engineering models are really good at figuring out mechanism, they're not highly accurate enough for biomedical applications. Whereas machine learning methods, are highly accurate, but it's really hard to understand how they function. So by sort of tying them together, we're sort of getting the best of both worlds, sort of getting mechanisms and also predictive power. So one key tool that we've been using is what's called as genome scale metabolic models of different systems. These have been traditionally used by chemical engineers for metabolic engineering for producing beer or biofuels. And we've been applying this for biomedical applications for, in this case, for understanding metabolic epigenetic interactions. And so what these models have are all known biochemical transformations that happen in a system. So the human model, for example, has more than 7,000 metabolic reactions. And it has, like thousands of metabolites and metabolic enzymes. And what it tells you is the relationship between what genes, these models tell you the connection between, like what genes encode what enzymes and what reactions are involved. Perfect. Yeah. So what genes encode different enzymes and what metabolites are involved in these reactions and how these reactions are organized into pathways and how they fit into the whole network. So we can sort of predict what happens when there are mutations in enzymes and how the systems level consequence of those. So I'm not going to go behind the math behind how we use this, but to give you a simple analogy, these models are very similar to how we use Google Maps. Now, in Google Maps, we provide Google with two inputs. One is the source and destination where you want to go. And then Google finds an optimal route based on another input, which is the constraint. You could find a different optimal solution based on cost or time involved. So depending on that, Google will tell you different optimal solutions. Similarly, on a metabolic map, we have inputs and outputs. So the cellular metabolism takes in dietary nutrients and then converts them into key biomass precursors like DNA, proteins, amino acids. So, and we have constraints. Just like in Google, we have minimizing cost. In the metabolic map, we're either, trying to be most efficient as possible based on thermodynamic constraints. And we also have constraints based on, protein levels, gene expression. So we can use all those physical and biological constraints to figure out what the optimal set of metabolic reactions are in a specific cell at a given time based on these different constraints. So now using these tools, now we've been able to predict potential routes that cells would take to convert different dietary compounds to produce key epigenetic precursors. And so we can use this to say what happens when there's a mutation in enzyme, changes in diet, and their epigenetic consequences. So just to give a simple example of how we use this to predict metabolic enzymes, which when mutated or perturbed can impact the epigenome. So now what we found is that there was like a small set of enzymes, when they're perturbed, do not have a strong metabolic phenotype. They don't affect the growth of the cell, but they impact, in this case, histone acylation. And the surprising thing is that the model predicted many known enzymes that affect acylation, but also new ones that are well known to be overexpressed or mutated in various cancers. And we found that in addition to the metabolic effect, this perturbation could have an epigenetic effect as well. And we've also taken this a step further to understand how the metabolic state could influence the potency or sensitivity to different H-stack inhibitors. So H-stack inhibitors are drugs that disrupt the epigenome, specifically acylation, and they've been used for treating inflammatory disorders, neurological disorders, and there are 4 different FDA-approved drugs, H-stack drugs out there. And one experiment that we did was, both computationally and experimentally, is we took cells, the exact same cells, and we treated them with the same H-stack drug called vorinostat, which is FDA-approved. The only thing we changed was that we added different metabolites to these cells, like glucose, fructose, succinate, and so on. And then we asked, how does the presence of these metabolites change the response of these cells to this drug, right? And what we found was that the metabolic environment has a big impact on the potency of this epigenetic drug, right? So we found that some metabolites like glucose, mannose, maltose make the cells more sensitive to this H-stack inhibitor, whereas some metabolites make the cells less sensitive. So these antagonize with this H-stack inhibitor. So this is really striking because it's an epigenetic drug, but the presence of metabolites can change the potency or cellular sensitivity to this drug. And the cool thing was that using our model, we can predict which metabolites would enhance sensitivity based on how they support histone acylation. So some metabolites increase acylation and they increase sensitivity to this drug. Others reduce it and they antagonize with this H-stack inhibitor. And so we've applied this to a variety of contexts. To give you one example, we've used this to understand how a high-fat diet could change the epigenome and potentially turn on pro-cancer genes in a breast cancer model system. So in this study, what we did was we took, in collaboration with breast cancer lab, took primary memory cells that are not transformed, and then we fed high-fat diet to these cells. And using our model, we were able to predict what are the epigenetic marks that would change and the metabolic pathways that will be affected. And what we found was that many of the genes that are turned on due to these epigenetic modifications were involved in pro-carcinogenic signaling, like MAP kinase pathway or NOTCH signaling. So this sort of tells you how changes in diet could influence metabolic pathways and how they could influence epigenetic modifications and increase disease risk. We've done this in fatty liver disease model systems as well. We've also applied this in stem cells, where we've shown that if you take even two similar stem cell states, in this case, we took a naive stem cell and a prime stem cell. These are embryonic stem cells, just few days apart in development, and we're able to use these models to tease out nuanced metabolic differences between these stem cell states, and then we're able to identify which pathways support distinct epigenetic marks, which influence the cellular identity of these stem cells. So far I've been talking about like metabolic epigenetic interactions that have been previously discovered. We've been using these models to simulate known metabolic epigenetic interactions. More recently, we've been developing machine learning methods to discover these new metabolic epigenetic interactions, so that we can identify how hundreds of metabolites in the environment or in the cellular environment could influence epigenetic modifications. So we've been taking data from hundreds of cell lines, for which we have both metabolic measurements and epigenetic measurements, and we're putting them together using machine learning models to uncover complex relationships between metabolites and the epigenome. So just to give you an idea of how this model looks like, is that we've found some really interesting connections between various metabolites and very specific histone marks. Rather than just saying metabolism affects histone acylation or methylation, we can focus on specific histone marks and identify which ones are highly sensitive to metabolism. For example, H3K4 acylation doesn't have too many metabolic interactions, whereas H3K9 has a ton of, is highly sensitive to metabolic state. So, for example, methyl nicotinamide here, is as antagonistic with acylation, but it's synergistic with various methylation marks. So now we can specifically point out when certain metabolic perturbation, how it would affect specific histone marks in itself. And we've been able to create these complex network diagrams, and we can link metabolic changes and how they together change different histone modifications. And we can also extend this to epigenetic drugs that perturb these histone marks. So showing a small snapshot here where we can identify metabolites that either synergize or antagonize with these epigenetic drugs. All right, so far I've talked to you about the metabolic epigenetic interactions at the molecular scale, but I would say one big challenge overall for this field is that how do you now link in high throughput these molecular level changes, the changes that are happening at an organ level or at an interindividual level, right? So the models I've talked about sort of link the molecular fractions and we build metabolic networks, combine these networks with epigenome, and we can predict cellular scale outcome. But moving that beyond to organ or interindividual level is like a big challenge for this field, right? And I think it's not unique to the gut-brain axis. We wrote a review where across various biomedical fields, the data or experiments are sort of siloed in different multidimensional space. So we can imagine that most biological experiments sort of measure certain components, right? It could be proteins, metabolites, and they usually measure that at a certain time scale or at a certain spatial level, right? And the key challenge for biology is, or for this field now, is to link these silos of data, that are sort of restricted to different time and space, and come with more models that are multi-scale sort of. And so that's the key step we need to sort of link these higher order interactions that we observe between diet and behavior or diseases at the macro scale to molecular level metabolic epigenetic changes that we measured in the lab. So it's sort of like an abstract idea. I want to give you one example of how we could do this using computational tools to link across scales. And so this one study we've been doing in collaboration with Gene Robinson's lab at UIUC, where we've used honeybees as a model system for studying this molecular basis of behavior. And so many of you know that honeybees colonies as a division of labor, right? There's a queen bee and there are worker bees that carry out the task in the hive. And the key thing here is that all these behaviors are driven by diet. And if the larvae are fed royal jelly, they become queen. And depending on the diet they receive, they become workers or other soldiers or nurses. And so diet plays a key role in deciding this behavior, but we don't know the underlying molecular level regulators that control this. And so using a combination of machine learning methods and high throughput experiments, we've been able to dissect the circuit that underlies this behavior. So what we did was, we used barcoded bees to quantify the behavior of individual honeybees. So now using deep learning, machine learning algorithms, we can sort of figure out what behaviors each bee has. Some might be nurse foragers that go out for food. Some might be nurse bees that stay in the hive and nurture the larvae. Some are soldier bees, that protect the hive. And some are generalists, surprisingly, that actually do multiple tasks at the same time. And then now we can connect these macro level behaviors to the molecular level by using transcriptomics and epigenomics data that have been done on the exact same bees. And we've been able to connect or discover transcription factors and other master regulators that are predictive of different behaviors. Now, some regulators are predictive of nursing behavior, some are predictive of foraging, some are predictive of, guards or soldier behavior. And we've been able to build another third machine learning model that sort of links these transcription factors to their target genes in the brain. So now you can see that we sort of get a mechanistic connection from how metabolic changes can trigger hormonal changes. And many of these transcription factors are responsive to hormones like juvenile hormone, insulin signaling that sense metabolic status. And these transcription factors turn on target genes that then predict behavior. So using like combination of different machine learning methods, we've been able to sort of connect, these axes, moving from the macro scale behavior, dietary changes, to molecular level changes inside the brain of honeybees. So to summarize, talked about these metabolic epigenetic interactions and how they underlie several gut-brain interactions at the macro scale. And then I talked to you about how we can use these genome scale metabolic models to simulate this complex interaction between metabolism and the epigenome. And we also been able to use machine learning to discover new interactions, how novel metabolites that have not can influence different epigenetic marks. And finally talk about this challenge in biology of integrating different scales, data sets, and how we can use a slew of machine learning tools to link across scales. And I think overall, this will help us move towards precision medicine using machine learning, where we can link a person's genetics, diet, and environment together to come with personalized medications or dietary changes that can enhance health. Yep. That I would like to thank you all for your attention and my funding source.
Well, that was an absolutely awesome way to begin this workshop. Again, we have 3 speakers this morning. We'll have everyone speak, and then we'll take questions at the end, both from the audience and virtually. It's a real privilege to be able to introduce our next speaker, Dr. Kevin Tracy, who is a professor of molecular medicine and neurosurgery at the Donald and Barbara Zucker School of Medicine at Hofstra Northwell University. He's also the president. He's also the CEO and the Karchus Family Distinguished Chair in Medical Research at the Feinstein Institute for Medical Research. Dr. Tracy received his undergraduate degree in chemistry from Boston College, his medical degree from Boston University, and then trained in neurosurgery at New York Hospital Cornell University and pursued additional research training at Rockefeller. He subsequently moved to the Feinstein Institute, where he's rapidly ascended to very senior leadership positions. Dr. Tracy is an inventor of more than 120 patents and author of more than 400 scientific publications. His laboratory discovered the molecular and neural mechanisms for the reflexive control of inflammation, now termed the inflammatory reflex, which has led to the first clinical trials of neuromodulating devices for the treatment of a wide range of inflammatory disorders. Dr. Tracy, it's a pleasure to have you as a member of this session this morning.
Thank you very much, Dr. Chakov. It's great to be here. And I want to thank the organizers for this terrific opportunity to discuss a field which I think is at a tipping point. I think this is a very timely meeting at a very important time, where literally decades of work in multiple fields, as you brought together so well in your opening remarks, Doctor, these fields are converging at a rapid rate, which is going to, I think, create an explosive new knowledge base, which is exciting in its own right. But as someone who's worried about trying to invent things to help people for my whole professional life, I'm equally excited about the opportunities to see this knowledge translated into useful therapies for millions of patients who need it across a range of syndromes. So that's a tall order. That's a big space to introduce a 10-minute talk. But what I will do is try to focus on the vagus nerve and what we've learned about it as a way, if you will, to study basic mechanisms to support what I said, but also to serve as a guidepost for strategies to provide new therapies. So what is the vagus nerve? Vesalius in the 16th century sketched it out as I like to say, it makes the human being look sort of like a jellyfish in this picture. But what it really is it's a principal conduit connecting the fundamental primitive brain, the brain stem, to essentially all the organs of the body. These are the organs that you don't think about in day-to-day life, but keep you alive: your heart, your lungs, your pancreas, and particularly for this meeting, the GI tract. The vagus nerve is described in scientific publications and in the lay press as the vagus nerve, singular, but there's two of them. It's a paired structure. And within each of the two pairs of the structures, there's actually 80,000 fibers. So the proper way to think of the vagus nerve is as 160,000 nerves. That would be a better way to think of it. And these nerves transmit information up and down from the body's organs to the brain to create the ability for the brain to maintain homeostatic function in all the organs. And it does this through reflex circuits. And physiologists, primarily in the 20th century, figured out in pretty good detail how vagus nerve reflexes control the cardiovascular system, heart rate, blood pressure, respiratory rate, insulin secretion, GI function, and there's a much longer list. And this was very well understood, but in the early 1990s, my colleagues and I, through essentially an accident in the lab, started thinking about whether it was possible that the vagus nerve was also controlling the immune system. And this seemed like such an obvious thing in retrospect. We went to the literature, spent many weeks combing the literature. So somebody must have thought of this before. But we just could not find evidence of that. And so what we've done essentially for the last 25 years, my colleagues and I in the lab, is study the mechanisms by which vagus nerve fibers can control specific aspects of inflammation. And it's funny, but it was 20 years ago that we proposed this model. And the model is simple and complex at the same time. So the simple aspect of the model is that, as I just told you, the vagus nerve has 160,000 fibers which communicate in both directions. 80% of them are sensory. That means they're capable of responding to changes in the body's environment and informing the brain about these changes. We proposed that, and it was known that sensory vagus nerve fibers, even in the 1980s and 90s, that sensory vagus nerve fibers could react to cytokines like TNF and IL-1 from the immune system. We proposed that perhaps the incoming information about the presence of inflammation could activate outgoing or motor inflammation, which could in turn modulate or even turn off the inflammatory responses. We also suggested that this wouldn't be limited to the vagus nerve because the incoming signals could also activate outgoing signals through the sympathetic nervous system, which could either increase or decrease inflammatory responses. That was known at the time. And the sensory input could also activate hypothalamic pituitary responses that could lead to the production of hormones like glucocorticoids, which could also increase or decrease inflammation, depending on what the adrenal gland was doing and what kind of inflammation it was. Well, the reason I'm here today is because this turned out to be true. And I'd like to give sort of an update on what we've learned in the last 20 years. So on the sensory side, it's fascinating because you can think of modalities or future diagnostics that are like the submarine listening to the undersea cable and decoding the telephone calls and the internet signals. And we've begun some work in that space using, with my colleagues, Chad Boughton and Theo Zanos and Shangita Shivan. And we've been using machine learning to record the vagus nerve of a mouse. It's not trivial to do. The mouse vagus nerve is about the size of a human hair. But when you look at what happens when you inject cytokines in that mouse, you see some very interesting things. So you see clusters of nerves in the time domain that are firing together in yellow that are sort of, don't matter if the mouse has TNF or IL-1. But look at the orange cluster too. Those neurons start to fire after we inject the mouse with IL-1, a cytokine. And they don't seem to change when we then inject the mouse with TNF, but a new group of neurons begin to fire in response to TNF. This raises the question or points to a future where we'll be able to continue to refine these modalities and actually decode the inflammatory condition. You can do this with glucose. We've done this with glucose and published on that. The metabolic condition or the dietary condition. All of this information is embedded in these vagus nerve pathways as electronic signals that can be measured. The challenge, as we just heard, will be one in machine learning and decoding, but it's doable. I'm convinced it's doable. Once the signals get into the brain, they're they are forwarded through brain networks into specific brain regions. The origin of the vagus nerve that sends the signals back out to the body is called the dorsal motor nucleus. And that's what you're looking at here is the dorsal motor nucleus of a mouse. In blue are the nuclei, are the cell nuclei of this nucleus. In red, the cells that stain red are expressing choline acetyltransferase, which means they're cholinergic neurons, which means they use acetylcholine as the neurotransmitter. And in green are the cells that we've, and this mouse is a transgenic mouse that expresses channel rhodopsin. This is a light-sensitive channel, and it's linked to green fluorescent protein here. So, or yellow, but it looks green in the slide, yellow fluorescent protein. So in the merged yellow that you're seeing, these are cholinergic nerves that are sensitive to laser beam light. And when the laser hits these neurons, they fire. And we know they fire acetylcholine because they're cholinergic nerves. And so a brilliant, now graduated, former graduate student, MD PhD student Adam Krasel and Sangeeta Sivan shined these laser beams on this region of the brain. And we were able to follow these signals from the brain stem of these mice, activating just a few hundred neurons, we estimate at most. And we know exactly what the neurons are. They're cholinergic and they go down the vagus nerve. They end up traveling into the spleen. And what you're seeing here is a nerve in the spleen, labeled in orange, that is within a few nanometers of a green cell. And the cell is green because it's a T cell that makes acetylcholine, believe it or not. So 1 to 2% of the T cells in the spleen can make acetylcholine. And the acetylcholine that's released by these T cells is actually the end signal to the immune system in the spleen that make the cytokines. And so when these T cells receive input from the vagus nerve, starting in the brainstem with a laser beam, they release acetylcholine, and the acetylcholine in turn shuts off the production of cytokines during inflammation. And so if you know that, then you don't need to put a laser beam in a human being. You can put in a neuromodulating cuff on the vagus nerve of a human. And so knowing this in the 1990s, we started first in mice and rats, which is what you see here done by Ludmila Borovikova. So these mice make a lot of TNF in the second bar because the sham mouse has been injected with LPS, which is a bacterial toxin, induces the production of large amounts of cytokines in this mouse or rat. When we cut the vagus nerve, you see the animal made even more. VGX is vagotomy. It's like cutting the brakes on your car. The immune system races out of control because it's lost this inhibitory circuit that normally turns off cytokines. And when we electrically stimulated this circuit, in VGX plus stim, you can see that the cytokine production is blocked. TNF is a major mediator of shock, low blood pressure. And when we measured the blood pressure in those animals, they were protected from shock. So putting this together with a lot of other work from dozens of laboratories and hundreds of people, I started a company. In my disclosures, I co-founded a company called SetPoint, which ultimately performed a clinical trial of this question in Europe using what I call a Gen 1 or Generation 1 nerve stimulator. It was off-the-shelf vagus nerve stimulating technology that was reprogrammed to hack into these neural circuits in an optimal way. This study was led by Frida Koopman and Paul Peter Tack. And this is important because this device has been implanted in more than 200,000 people worldwide, and it's safe. It does not have a black box warning, as opposed to virtually all the drugs that are FDA approved to treat rheumatoid arthritis in the United States. It does not have any evidence of immunosuppression. And at this time of this study, it was unknown whether it could actually be effective in activating the inflammatory reflex in patients with rheumatoid arthritis. When we first implanted this device at Northwell Health in New York and turned it on, we were very pleased to see that the activation of electric current flowing in the vagus nerve, as predicted, inhibited the production of TNF in humans. And this encouraged us to go on and study patients for 84 days in the first cohorts. Cohort 1 patients had severe rheumatoid arthritis that was refractory to steroids and methotrexate. Cohort 2 patients were also refractory to biologics and advanced therapeutics. And as you can see, there was a significant clinical response by implanting a vagus nerve stimulator in these patients. This mean change in DAS28 CRP means the mean change in disease activity score based on counting 28 tender swollen joints in these patients and adding in a factor for serum CRP levels. This is a very robust response in any first round clinical trial of even 17 patients. Since then, you can actually see that when the treatment was turned off in what's called the treatment hiatus, the patients got worse. The good news is when we turned it back on day 56 and followed them for 2 years, which is not shown here, they improved again. And many of these patients have had lasting benefit. And some of these patients are disease-free, taking no drugs, just the vagus nerve stimulator. So where are we? This is a future-looking conference, and this is a future-looking topic. To create some context for the future, every year on the planet Earth, about 160 million people die. Approximately 40 million of them die of inflammation every year. Inflammation being defined here as diseases which are either started by or made worse by or accelerated by inflammatory mechanisms. These diseases, according to the WHO, commonest killers are cancer, cardiovascular disease, dementia, neurodegeneration, diabetes, and obesity. Understanding the ability to interact through neural circuits to control inflammation is being actively studied as I speak in all of these conditions. And in many of these conditions, there's not only interesting preclinical data, but there's in some of these conditions, there's extremely important, you're going to hear tomorrow from Dr. Lim, some extremely important clinical data, as in diabetes, for instance. This space of bioelectronic medicine is an opportunity for the future because it brings together, again, as Dr. Chekhov said in his opening remarks, 3 fields that normally haven't been intersecting. And the first is the molecular therapeutics or the molecular biology of disease mechanisms. I call that the target. The second is the neuroscience or the neural circuits that can be identified mechanistically to control those targets in situ. And the third, of course, is the bioengineering and the device, which we can now, of course, design devices that can hack specifically into neural circuits to control those targets. I'm very excited about the future, very pleased to be here today, and thank you.
It's a pleasure to invite our last speaker for this morning session, Dr. Mauro Costa Mattioli. Dr. Costa Mattioli is a principal investigator of the Bay Area Institute of Science at Altos Laboratories and an adjunct professor in the Department of Neuroscience, the Cullen Foundation Endowed Chair in Neuroscience and Director of the Memory and Brain Research Center at Baylor College of Medicine. Dr. Costa Mattioli received his undergraduate degree at the University of the Republic in Uruguay, his MS degree from the Pierre and Marie Curie University in Paris, and a doctorate from the University of Nantes, with additional postdoctoral trainings at McGill University. Dr. Costa Mattioli has elucidated central mechanisms underlying neurological dysfunction, including the mechanisms by which specific microbes in the gut modulate brain function and complex social behaviors. His awards include the International Eppendorf and Science Prize in Neurobiology, the Searle Scholar Award, the International Society for Neurochemistry Young Investigator Award, and the UCSF Presidential Award. Dr. Costa Mattioli, thank you very much for joining us.
Thank you, Elliot, for the presentation, introduction, and it's been a pleasure to work with you, Kenya, Dayan, Kavita during the last few weeks to help to put together, which I think is an incredible workshop. So there is a new movement in neuroscience, as you have heard, which is growing in momentum for this idea that essentially we can modulate high cognitive functions by structures outside of the brain and by cells, which are non-neurons. A concept that if we were to be talking 10 or 15 years ago, it would be actually inconceivable. So in my lab, we are interested in understanding the mechanisms underlying neurological disorders. And when you think about the brain, traditionally, the brain or behaviors, which are modulated either by environmental or genetic factors, invariably, we think about the brain. But we and others, we have shown that microbes in the gut could modulate the brain in a very powerful way. And these have added a new view, more holistic, new integrative, by which essentially behaviors can be controlled. The story that I will tell you today is a result of a serendipitous discovery initiated by a very talented postdoc at the time, Shelley Buffington, who was interested in autism. And in the lab, we were interested in genetic forms of autism, autism that is characterized by three major symptoms: social deficit, communication problems, and repetitive behaviors. And as you know, unfortunately, despite increasing the incidence of autism, today there's no treatment that essentially can alleviate any of those symptoms. But instead of the genetic forms of autism, Shelley was interested in maternal obesity. And the reason why she was interested in maternal obesity, because there is epidemiological evidence that maternal obesity increased the risk of autism in kids. So in order to study this process, we created a model where moms were given a high-fat diet. And of course, we put a male and we measure the behavior, and the moms, of course, they gain weight. And we measure the behavior of the offsprings. And we measure a variety of behavioral paradigms. For the sake of simplicity, I will just describe a task, which is a social task, in which the animal has to decide whether to interact with a mouse, with a cage where there's a mouse, or in a cage where there is essentially no mouse. And if you are normal, and if you are a mouse, you spend most of the time interacting with the mouse, as you could see here. But the animals that come from moms which are obese, they cannot distinguish whether there is a mouse or not in the cage. Of course, they can distinguish, but essentially, they decided to spend equal time in one compartment versus the other. And this is a clear indication that social behavior is actually impaired. But here's where serendipity struck. 3 weeks, when we wean the animals, and we mix one of the maternal high-fat diet animals with 3 of the control animals, what we discovered that the behavior of the animal that was sick, the behavior of the animal that have a social problem, actually was improved. And we scratch our brain because, and of course, we repeat that experiment, and invariably of the ratio that you can use 3:1, you can use 2:2, you can use 1:3. Invariably, every single time that you put a mouse which is social deficient with a control mice, their behavior is actually correct. To make a long story short, I will tell you what happened here. Because what happened is the diet, maternal diet, is going to eliminate a microbe in the gut, in the offspring, and that microbe is required for social behavior. And because animals are coprophagic and they eat each other poop or feces, when you put them together, that microbe that is missing in the red group is actually provided by the control animals. And when we go and we sequence the microbial ecology in the control animals and the maternal high-fat diet animals, we clearly see a difference. Actually, we don't need to do behavior any longer. By just looking at the microbial community, we can determine whether an animal is normal or has social problems. And you remember the animals that they were socially normal, their microbiome is completely corrected. But this is an association, not a causal evidence. In order to demonstrate that that was the case, we study animals that have no germs. Those are animals that have no microbes. Those are called germ-free mice. And when you look at those germ-free mice, like the animals that come from moms which are obese, they are socially impaired. But if we transplant the feces from the control animals, we are able to reverse their behavior. But if we transplant the feces from the animals that lack that microbe, because their microbe is not there, social behavior is actually not reversed. And you should know, actually, that the microbiome not only controls social behavior in mice, but it does it in bees and also in zebrafish, demonstrating that this must be an evolutionary conserved process. Now, in order to understand what was the microbe that was missing, we sequence deep and we identify a bacteria strain which is called Lactobacillus reuteri, whose abundance is actually dramatically reduced in the animals that are socially impaired. And incredibly, when we put that bacteria, we are able now into the animals that they are socially impaired, we are able to reverse the social deficit. Yes, it's the addition of one bacteria, but not any one, because if we add a cousin of Lactobacillus reuteri, Lactobacillus johnsoni, we fail to reverse the social deficits. Some of you may wonder this might be a specific effect from this maternal obesity model. But we wonder whether that was the case. And we use a variety of preclinical models of autism, mostly genetic ones, including animals that have a mutation in SHANK3B. And those animals, they are socially impaired. But now if you were to add that bacteria, you are able to reverse their social deficits. And actually, we have done it in 6 preclinical models, and many investigators around the world, actually, they have extended and actually reproduced our findings that actually emerged only in 2016. Now, mechanistically, what we discover, what the bacteria does is increase a hormone which is called oxytocin, which is called the social hormone, in a particular brain area, which is called the paraventricular nucleus and the hypothalamus. There is a connection between this area with a reward area, an area that sends reward. And what we discover is that if we put 2 animals to socialize and we put an electrode in the reward area, those neurons fire it in a similar way that they would fire it if you were to give to the animals cocaine or any other drug of abuse. However, if the bacteria is not there, the circuitry is actually impaired. And as a result, when you put 2 animals to socialize, those neurons, they don't fire it. And we believe, at least, that this might be a reason by which essentially animals or perhaps individuals don't socialize, because they have absolutely no pleasure associated with it. We also identify specific circuits, and if we were to remove the receptors in those reward neurons, now the animals are social, and as you would expect now, as Kevin pointed out, if you were to block that, the mechanism underlying the process, now the bacteria fail to reverse the social deficit. And when you look at the excitation or the changes in synaptic activity now that typically take place in response to social behavior, now in animals in which those receptors are not there, you can essentially, if you fail to elicit that response. You may wonder, this is a specific feature of only mice. The answer is no, because in children with autism, social interaction actually seems to be no rewarding according to imaging studies. Now, this field is a nascent field, and probably the aficionados in the field, they have learned about a particular microbe that modulate a particular behavior. But here are, according to us, which are the necessary steps. The microbe should have a gene that produces a molecule, and it has to be a signaling mechanism. And I have no time to show you, but yes, Kevin, the vagus nerve is responsible. This is the structure essentially that sends the signal. We've been talking with Theo. We're trying to figure it out whether we can decode how that signal is actually transmitted into the brain. Sean Donnan in my lab try to identify, and Shelley Buffington, they try to identify where is the molecule underlying this. And when we look at in another model of social behavior or social dysfunction, in a genetic model of social dysfunction, what we discover is that there is a specific metabolites which are essentially corrected by this microbe. If you look at the overall metabolic profile of the gut, is different between green and blue, between wild type and diseased animals. But if you were to treat with the bacteria, you failed to completely reverse the metabolic profile, but nonetheless, you are able to reverse a specific metabolites. And these metabolites from the biopterin pathway that essentially are required to synthesize a variety of neurotransmitters. And what Sean found that if he was to give these to animals now in a bacteria-independent manner, what the bacteria induce, now you're also able to reverse the social deficit and the associated changes in synaptic function. Now, the point that I want to make, that the manipulation that we have in here, either biopterin or the treatment with the bacteria, sorry. Oh, how do I go back now? Okay, only is able to reverse the social deficit, sorry. Because when we look at, say, hyperactivity, now hyperactivity is not changed. There's a specific manipulation that reverses the social deficits. And as I always said, until the last 6 months, I will be entirely happy if everything that I told you today has happened in animals, because it has opened a completely new way for our lab to understand biology. But as Kevin pointed out, the advances are going forward in an incredible pace. And together with Luigi Mazzoni, University of Rome, sorry for the typo, and Antonio Hardan, University of Stanford, during the pandemic, a clinical trial was performed in 20 kids, in which this double-blind placebo, and then they were given the treatment, which is Reuteri. And as you could see here, social behavior, not the severity of the disease, not any of the other behavioral abnormalities, but social behavior is actually improved. Now, what gives strength to the field is your orthogonality. That is like other people should come with the same conclusion. And I was amazed to see, this is about 20 kids, double-blind placebo. These papers actually under revision. But incredibly, I was not aware until 2 weeks ago that this paper was deposited in the bioarchives of another trial with the same bacteria, but also with the same results. Now, if you were to look between these two bars, animals, children that they are socially impaired, if they were to be given these bacteria, now you are able to improve their social deficit. And in this particular case, they even look at oxytocin in the plasma, and they found that oxytocin is actually increased. So what is the take-home message that I have for you is that the precision microbial intervention target a social reward circuit that is deficient in mice with autism, increases oxytocin in the brain and biopterin in the gut, reverse the social deficits in a wide range of models, at least 6 preclinical models, multiple laboratories, and improve social behaviors in children with autism. And these are two independent trials. Of course, this needs to be extended. The only thing that we are seeing here is a signal, but we are incredibly excited because, as you know, for the main core symptoms for autism, we don't have a treatment. And we cannot have a treatment that have any toxicity whatsoever because we will be treating children. And of course, I didn't have time to tell you, but this bacteria is super safe. It has not negative effects whatsoever, even in infants. So thinking about the future, we need to better understand the mechanism that underlying this. And I think Kevin didn't have time to tell you about his story about TNF-alpha and how he ended up with the vagos and, you know, and antibodies, and I think it is understanding of the mechanism that would allow us to essentially develop better medicines. Perhaps we could think about a new generation of probiotics or even metabolic products. Ultimately, the way that I see a microbe is a bag full of chemicals. But the chemical space that those microbes have is incredibly vast. Could we synthesize specific communities to promote brain health? If we know what are the microbes which are good, and we know which are the microbes which are bad, we also can develop perhaps food therapeutics in a way to conserve the good guys and get rid of the bad guys. Now, I'm incredibly fortunate by the people. I think I mentioned the main people during my talk, funding agencies. And now I just want to spend 10 seconds to tell you that those experiments in reality were like sort of not easy, but the experiment that I'm doing here is a big one because an experiment with myself, probably the biggest experiment in my life. This is a new model of scientific inquiry. We try to mix the best of academia and the best of industry with the goal of pursuing big questions with team science, scientific openness, and scientific freedom. And we are like about 30 of us that we were in academia, very well established, that would have decided to join these what we hope could serve to transform hopefully science and medicine. And I will be happy to take questions. Thank you.
That was a fantastic way to start this two-day workshop. Really incredible speakers. And I'm sure there are questions both from the audience, and I see at least one question up on the Zoom chat. I see Charles Gobert has his hand up. And actually, I'm not sure if people from on the virtual podium can hear you. Is there a microphone for, is it portable at all, or are we going to move it into the center of the room? Let me start, while that's being set up, let me start with a question from the chat for Sriram. How transient or permanent are the changes to the epigenome by metabolites? The individual is wondering to what extent our day-to-day or week-to-week dietary choices impact long-term risk of disease.
Yeah, that's a great question. So it depends on the strength of the metabolic perturbation. Right? I mean, I think the subtle metabolic change, maybe it's not, the enzyme is sensitive to the relative levels of these metabolites, and their activity might not change. There's a subtle metabolic change, but if it's a huge bolus of sugar or high fatty acids, then that's enough to change the intracellular metabolite levels, and that would change the epigenetic enzyme activity. So it depends on the strength of the metabolic perturbation. It could be small and strong, or it could be slow and long. It depends on the metabolic perturbation and how, and that influences the epigenetic state. Yeah. So, but it's a good question. It is context dependent.
Can I add to that question?
Yeah.
Are the changes reversible?
Oh yes. Yeah. I mean, if, for example, you could think of a yeah, you could think of maybe a combinatorial. For example, you have high fat diet that maybe increases histone acetylation, but then you could think of a second metabolic change that could potentially reverse that. People haven't done that, but I think it's possible.
Charles.
Okay, so I have a question that is a general one that is to all the speakers, and that has to do with specificity and selectivity. So for Sriram, if you were expecting to have diet influence the state of methylation, say, of the histones, how can you get a specific gene effect by, or is this just a general gain control where all the histones that are methylated in the same way would be affected, and it would be sort of a more global effect? And for Kevin, if you're expecting the vagus, and so we know the vagus has a diversity of projections to a number of different brain regions. And if you expect that there may be a specific set of vagal afferents that affect inflammation, how would you be able to achieve that effect on inflammation by vagal stimulation, where you might be activating too many of the targets of the vagal nerve? And for Mauro, the lactobacillus So it's known that the small intestine has a number of different categories of cells that are innervated differentially by the vagus. And so what is the product of the bacillus that it will only influence that subset of the cells in the small intestine that project to the PVN and affect oxytocin release?
Kevin, you want to?
I get to go first. The question you ask is an area of clear importance and being pursued by many. Specifically, in the context of inflammation and cytokines, it turns out that the fibers that project from the dorsal motor nucleus to the spleen are exquisitely sensitive to low thresholds of current. So you can apply current to the whole bulk of the 80,000 bundle in the vagus nerve on one side, but by putting in a few hundred microamps of current, it's sufficient to activate the neurons that turn off the cytokine cascade, as opposed to laryngeal function or even cardiac function where milliamps are required. So we are not seeing significant side effects. That being said, your question is still $1 billion question in terms of understanding the brain network mechanisms. We and others, particularly Asia Rolls in Israel, Jeff Friedman at Rockefeller, and Charlie Zucker at Columbia, have either all published on or are doing genetic models of neuronal trapping. So you can literally treat a mouse with a nutrient, or in our case, a cytokine, and because of through genetic engineering with this trapping technology, you can label the neurons that fire only for a few minutes or hours after the administration of the agent. So you can then go back and use pharmacogenetics or optogenetics to stimulate just those brain neurons. And what we're finding is a few brain neurons activated by a specific cytokine can replicate the effect of the cytokine 2 weeks later in the absence of the actual cytokine. So there's clear proof that neuronal circuits in the brain report or respond to the change in the environment, activate a cassette of responses which can then reproduce that environment. It's really exciting research, and it is changing the way that we think about these neuronal immune interactions.
Maura?
Kevin, so does the vagus interact with the splenic nerve? So, but if you were to, no, it's not.
I've been saying that for simplicity today. The origin of the splenic nerve is in a ganglia called the celiac superior mesenteric complex. The vagus terminates there and controls the cell bodies in that ganglia outside the spleen.
And Sriram, did you want to respond to Dr. Gilbert's question?
Yeah, I would say I had the same question when I was listening to both of them talks too. So, but for the epigenetic part, and I would say normally in cells during normal function, the way plasticity happens is that these metabolic enzymes actually localize in certain parts of the nucleus. And so if you have these big metabolic chains, that it could have widespread epigenetic effect, but if these enzymes are localized in certain parts of the chromatin, then only those genes are, turned on or turned off. So that's how it normally happens. So if we were to do this, like therapeutically, I would say, diet would be a blunt tool. It might affect epigenome everywhere, but in combination with the pharmacological agent like, verinostat, where that localizes to a certain immune cell type, then using a combination of which then you can make it more specific. So yeah, diet overall would be a more blunt tool that could affect all cells and tissues.
That was a great question. And the box that I was unable to uncheck in my presentation is what is the gene in that microbe that drives the behavior. And we did a very low through, a very talented student now, a scientist in my lab, a very slow throughput screening, behavioral screening with different strains. And we discovered now that there are some strains of reuteri that are able to reverse the social deficits, and others they're not. They're both reuteri. And in between each other is about 200 genes which are different. And now we are in a position, I think, we narrow it to about 2 genes. When we have that, at that point, we can essentially use those 2 strains to determine what is being produced by the microbe and what is being activated. If you were to use two different strains with 200 genes, the difference that you have in metabolites is massive. Let's say if you were to use Reuteri and Johnsonite. So before to do that experiment, and because we need to reduce the noise, we had to get to the gene. So then we can use a normal strain and a strain that just lack two genes, or those two genes are inactivated, to get precisely to the mechanism at that level, which is essentially, which, as you pointed out, is missing.
And to be clear, did the two clinical trials use the same strain?
Okay,
Right?
so in the two clinical trials, in the second trial, they don't reveal, it's Reuteri, but they don't reveal what is the strain. In the first clinical trial, it's a tablet with two strains. And when we look at the effect of the two strains separately, only one of the strains is driving the behavior in mice. So for future trials, we believe that it would be beneficial. We believe that we are diluting the signal. I think it will be beneficial to use one of the strains.
All right. Is there a question?
Hi, first of all, incredible talks. My head's swimming with questions, but I'm going to limit to one for each of you if people indulge me. So, first of all,
Could you speak up a little bit?
Sure,
Sorry.
I'm sorry. Can you hear me?
For especially for those who are on Zoom.
Okay,
Thank you.
Great talks. For Sriram, I'm wondering if through your work you've been able to identify the behavioral circuits that you're working on, both the genes that are involved and the structures. If that work is, if you've identified them.
Yeah, we've found the genetics, gene regulatory circuits, but probably not the neural ones, so we've been focusing more on the molecular level. So we can identify specific regulators and targets for individual behaviors. So that part we can do.
Okay. And for Kevin, it was just so many thoughts here, but the one that popped in my head, and this is rather, I think, naive, but it suggests to me that this could be really important in how emotional stress affects the immune response, how placebo and nocebo types of effects could be potentially explained. I'm just wondering if you had any thoughts on that.
Yes, we have a lot of thoughts on that. And the issue comes down to, in order to have a mechanism oriented strategy to connect the pieces of neural signaling to behavior to molecular mechanism, you really have to address first diseases in which the molecular target is understood. Once you move into anxiety, depression, schizophrenia, you have the problem of not knowing what is the molecular basis of the disease. And so it's very hard to connect it to a neural circuit. That being said, FDA has approved vagus nerve stimulation for the treatment of depression and anxiety in the United States. CMS did not authorize payment for it, so it's not used, but it is used in Europe. And what you have in those cases, there's more data now from Europe than the US, is that there is a treatment effect in some percentage of patients with depression and anxiety from vagus nerve stimulation. It's not 100%, it's under 50% treatment response. And so the issues come down to what is the mechanism. If you knew the mechanism, you might be able to screen patients in advance of the surgery and then know more what you're doing. So in the case that we focused on inflammation primarily, it's because we understand the mechanism of monoclonal antibodies blocking cytokines in humans, and we can work up and down the mechanistic cascade from there. So that's the opportunity and the question.
Thank you. And for Mauro, I was wondering if you were able to identify from the human studies that you were doing, if you saw a better response from the children in terms of their age. The earlier they are, the longer they are on this particular bacteria treatment, if they had a better outcome.
Yeah, so just take it with a grain of salt. Our trial is about 20 kids. We don't think that we have power. The other is 7 and 8. The reason why I'm telling you to take it with a grain of salt, because we have just 20 kids. From those 20, we didn't see the kids who were high functioning were the ones who respond better. but it seemed to be, but we don't have the power to make that claim, right? So, but this is extremely important, you know, and in the next trial, obviously we will focus on high functioning, but also we will, after the trial, we will look at responders versus non-responders, and we will look at the microbial ecology. Could it be essentially that there is something that we can determine before we do the treatment, but just look at, you know, at the microbiome of the children that could predict whether they will respond or not. But again, I think it's early days when we have, you know, the number of patients that have been used for vagal stimulation. I think we will have a better number, but with 20 or another trial, you know, 7 and 8, I don't think that we are in a position yet to answer that question with, you know, with strength.
Good question. Yeah.
Todd Coleman online has a question, and we're going to bleed over a bit into lunch.
Good morning, everyone. Sorry, I'm not not with you in person. I really enjoyed all the talks, and I had a couple of questions. First, a comment, Sriram. I started my career at Illinois, Urbana-Champaign, and got to know Gene Robinson very well. So when you discussed the work on the bees, that was really exciting. I had a question about the work in the bees, and you talked about how we know there the diet really affects social behavior. If we wanted to translate some of these types of stories that you're telling with bees to explore if analogous modifications can be made in humans, what do we know about the gut-brain axis in bees? You know, to what extent is there and what is not, from which those findings have the potential to translate? Or have you guys put thought into that?
Yeah, on the biological side, I guess, I mean, the underlying molecular circuits are similar. Like we've done some studies comparing, you know, like the underlying metabolic response for aggressive honeybees and how their metabolism is very similar to many of the neurological disorders we see in humans. We have done some evolutionary comparison of those circuits, and we do find some deep conservation there. And I guess on the computational side, I mean, the tools in theory could be applied to humans. You know, like we have precision measurements of humans, you know, the fitness trackers, or people measure their diet. So I could potentially see how we could combine those behavioral and dietary measurements together with genetics to do that, make that possible. But once again, all the honeybee data is done in one lab, everything is clean. It's easier to integrate them, whereas human data is going to be more messy. So I think the circuits might be similar, I mean, the molecular level, because there is some conservation, but putting them all together, it's going to be a challenge.
Kevin?
So I can't speak to honeybees, but Alejandro Abele and his colleagues have mapped in C. elegans, of all things, a cholinergic circuit, a very simple cholinergic circuit that targets the primitive gut immune system of C. elegans and turns it off. So I call that the vagus nerve of C. elegans, although I'm probably not authorized to name it at this point.
That is awesome. That is a great. Thanks so much, Kevin. And I had one question for Mauro. I really enjoyed. You talked about the Lactobacillus rhudieri. You already clarified one thing for me, which was that there are off-the-shelf products already that have that are probiotics that have L. rhudieri as one of them inside, and a lot of pediatricians recommend them for children. But what you said about how the details of the strain really matter, I found that to be quite fascinating. So we'll be tracking that in more detail. My bigger picture question was, one thing I was curious about is you focus on Lactobacillus rhudieri, but we know that all these microbes compete and cooperate with one another. And what came to my mind is that when you think about the amount, like suppose if this kid is going to take a pill, the amount that the kid is going to take, that might depend upon the status from the second it goes into the mouth before it is, you know, in the small intestines and where it starts to engage the gut-brain axis. And do people pay attention to how there's variability in the acids in the stomach or the duration of time that the stomach is processing things, and how that might alter the ecology, so that what's in place when it begins to go into transport into the brain could vary quite a bit because of the individual variability, not only in the gut, but what happens before it reaches the small intestine. Do you guys pay attention to that kind of stuff?
I think this is a great question. I think it's going to be highly unlikely that the microbe will colonize. But you know, the way that the trial was done, our trial, is we give the microbe twice a day. So the microbe will not colonize, but it's going to be metabolically active. Okay. But eventually, if there are conditions that inactivate the microbe, because we showed that when we kill the microbe, now the microbe is not effective any longer. So eventually, any condition that will kill the microbe in the gut, you know, probably is gonna add a variability to the study. But because we are giving it twice a day, we cannot measure. You know, you will have it every single time that you take it from the feces. Obviously, you will have an increase in the microbe.
Thank you. Those are my questions.
Diane, did you have a question?
Are we okay at going over the time a little bit? Okay, so fantastic presentation. And like the others, I have had a million questions right afterwards, but most people are also asking the same questions that I had. And I wanted to also comment, I think Mauro said at the beginning, 15 years ago, we wouldn't be having this conversation. One thing that strikes me about this is we're talking about gut microbes, we're talking about genomics, we're talking about epigenetics, we're talking about complex computation and machine learning, and we're talking about the brain and the nervous system as a holistic system that has to be understood to think of treatments for things. I mean, it's not like here's a drug and here's a receptor we're trying to, you know, antagonize. So I'm very much struck by that. My specific questions are on the neural side of things. I wondered if there was implication for vagal nerve stimulation in PTSD. I know you mentioned depression and anxiety, but do we know much about PTSD and whether or there, you know, we could see a future where PTSD could potentially be prevented in known situations that people were going into. And then I had a question for Mauro, which is in the autism spectrum disorder world, there's sort of this, there's a huge emphasis that people have been trying diets in their children, and there are all kinds of websites that the parents go to, and they look at these very specific, you know, almost religiously following certain diets that they are told. And I'm wondering if you've looked at any of that, even just anecdotally. Is there something that affects the bacillus that you're observing? Is there really any truth to these dietary anecdotes that may actually affect that specific microbe? Thank you.
Thank you. The PTSD is another area of active investigation. The last time I looked on clinicaltrials.gov, there were more than 100 clinical trials of vagus nerve stimulation. Many of them were for conditions, as I said before, like depression, anxiety, but also PTSD. The issue with doing the clinical trials now is twofold. The first is that we don't know the mechanisms, and if you don't know the mechanisms, you saw it, connecting the dots through oxytocin or through TNF. It bolsters the ability to do something, to change something if the trial's not working, if you know what you're doing and you know what the target is. So that's one opportunity, issue, problem in that space. The second is that the FDA has approved several different non-implanted vagus nerve stimulators to activate the vagus nerve. Now, the issue with that is it's not clear that they're all truly vagus nerve stimulators. So some stimulate through the ear, and I would properly say that they are TENS units that stimulate the nerves of the ear, one of which in most people is a branch of the vagus nerve. So technically you're stimulating a sensory branch of the vagus nerve. Is that the same as what I showed you in implanting a neuromodulating device, or using ultrasound, or using some other direct parameter where you know you're targeting the structure of the vagus nerve proper? To me, those are very different. Now, that being said, there's some very interesting data using TENS units in the ear. It's an FDA-approved therapy for opioid withdrawal, for migraine headache, and I'm forgetting one. Oh, I said, there's several FDA-approved indications, including opioid withdrawal and migraine headache. So it's an area of active interest. There are currently ongoing clinical trials using that approach for PTSD. I'll be very excited. Hopefully the day will come when the FDA will approve the surgical devices for rheumatoid arthritis, because I think that will open the doors for many additional clinical trials for inflammatory and CNS conditions.
And Mauro, did you want to, there was a question that was directed to you?
So a couple of things. So the first one is we haven't looked. And the reason why we haven't looked is because this bacteria has been eliminated in the population. Actually, I think it was in the 20s or even earlier. You can find them in natives. No, I know that you will jump and you will say, oh, that's why we have so many autism kids. No. It has been eliminated and actually has a sensor that it has like viruses which are integrated into the genome that they are sleeping. And when they sense stress, those viruses get activated and kill essentially the bacteria. Whether this is diet or actually, anxiety or depression, this actually remains to be seen. But let me just close with a romantic note. And because when we think about microbes, when we think about microbes, what microbes do is they hijack the cellular machinery to be able to reproduce or produce themselves. In the case of viruses, they use our machinery to produce viral proteins. I think what this bacteria is doing is hijacking the system that essentially regulates social behavior. But what better for a microbe is to promote the communications between two individuals. So then actually the microbe can be passed from, one generation to the other. So again, that's my romantic aspect. That's why
I thought that was an evolutionary aspect.
Okay, exactly. But yeah.
Thank you for a set of amazing talks. Catherine Cabrera from Lincoln Lab. I was wondering if any of you have thought about flipping it a little bit, and instead of thinking about medical countermeasures and treatment of psychological or behavioral disorders, can we learn from the microbiome what disorders may exist, particularly for things such as suicidal ideation or psychopathy or other kinds of concerns? Are there ways we can exploit analysis of the microbiome or the function of the microbiome to understand these invisible but concerning psychological disorders? Thank you.
Mauro, you want to take that on? I think you.
The field as a whole, for the last, I would say 10 years or so, they have tried to find associations with, let's say, autism with a particular microbial ecology, or PTSD, or Alzheimer's disease. Unfortunately, I want to say, and maybe some people in the audience may disagree with me, we have failed to identify a signature. Let's say in kids with autism, a large number of them, in some cases, they are reported 70% of the kids, they have gastrointestinal problems. But we haven't failed to find a signature. A signature is one that happens in Kevin's lab, that has happened in Elio's lab, that happens in Sriram's lab, that happens in every single lab. So what the field suffer is that Kevin find a signature, Elio find it in the same, not in the same kids, but in kids with autism, a completely different signature, and so on. And I think that this is inherent to the variability which is associated with the microbiome. Perhaps machine learning or other methods will allow this. Perhaps use biosensors would be another way. But I think we need to think again how we can detect those changes in a more reliable way to create predictive value. But I think that this is what we are at.
It is a fundamental challenge of causation versus association, and which came first, chicken or egg. But does anybody else wish to weigh in at all?
Just want to add that, for many of these observations people have made, like it's complex because there's a genetic component and the diet. For some people, the vagal stimulation might work depending on the genetics of the diet. Right? We should take that into account when doing these studies. So that's why there are these anecdotal observations on autism or others, because maybe it worked for one person because they have this right genetics and the right environment. For others, it didn't work. So I think putting all three together, then maybe we could do this reverse causation where, hey, for this genetic profile, this drug with this stimulation might work, but not for others. I guess that's what we should go towards, and we need data for doing that.
And just let me add one layer of complexity. And I think, so we have a study last year in which immediately when we think at a genetic disorder that impacts the brain, we believe that that gene that is missing is affecting a circuit or a molecule in the brain. And as a result of this, we have a behavioral change. But if the mutation is germline, so it comes from mom or dad, it's very likely that it's not just in neurons in the brain. It's likely that it's in every single cell of the body. Or if it were to be in neurons, for sure would be in the enteric nervous system. And what we have found, and other group with a different mutation has extended our findings, is that a mutation in the brain affects one behavior, and the same mutation, not in the brain, in the gut, change the microbial ecology. And because the microbial ecology is disrupted, you affect another behavior. Thus, if you were to treat this disease and you were only focused on the brain, in principle, you will be able to improve only one symptom. And the opposite would be true if you were just to treat the gut. So we need to perhaps in this holistic view, perhaps we should consider that, the two components could be treated.
I just want, question? Could you use I'm sorry, just because we have people on Zoom.
Fascinating talks. I had a question about perhaps reversing the dialogue of the exploration. And have you worked with behavioral specialists in looking at how behavioral therapy might change microbes in the gut and then predict a similar response that you're seeing when you change that microbe independently?
So there's fascinating behavioral approaches to the question of the relationship between vagus nerve signaling and inflammation. And it turns out if you look at families, cohorts of relatives of patients with rheumatoid arthritis that have genetic markers for being at risk for rheumatoid arthritis, but they do not yet have clinical signs and symptoms, that if you measure their vagus nerve activity indirectly by things like heart rate variability or other surrogate measures, that they have impaired function in the vagus nerve. So it's a behavioral strategy to say, is the failure of the neural circuits occurring first, which allows or then accelerates the progression of inflammation, which causes the signs and symptoms which come later. So there's a lot of interest in that backwards approach, and it's yielding important data.
That's a great question, and I don't know the answer. But just to follow up what Kevin pointed out and the question related to PTSD. It turned out that in 2000, a colleague of mine discovered that if you were to recall the pathological memory, if you were to recall it, that memory, which is pathological, became fragile for about an hour or so. And at that moment, if you were to have a way to intervene, like with vagal stimulation, you can erase, but you can only erase the memory that you recall. So what many people are doing now, they are trying to identify strategies to modulate the system, and they combine them with behavioral therapy. If you were to do that manipulation that disrupt the memory when there is no recall, you cannot erase it. If you do it after 1 hour or 2 hours after the recall, you cannot do it either. Because in this particular instance, I will have a hard time to believe that the microbiome will be at play because it's super quick. It's a window of about half an hour or an hour or so where you have to intervene. It's true If during that hour, in addition of the pathological memory, you recall your boyfriend will go too, and you will erase it. So, so that's why we need to have precise or precision behavioral therapies that I trust that combine with, let's say, vagal stimulation or other technologies will be, I want to believe, in some cases, in the case of PTSD, they can be more effective.
And because of this conversation, I now remember the one I forgot. It turns out the FDA has also approved in the United States, with apologies to my colleagues in Texas who did all the work, the use of stimulating the with a TENS unit in the ear and electrical stimulation of the branches of the nerves to the ear to enhance the efficacy and to shorten the timelines for recovery from stroke, from a cerebrovascular accident, and this turns out to be extremely important in accelerating the recovery from people with motor impairments. Assuming that the therapy can be delivered exactly as you heard in a time window that modulates brain plasticity. So yes, your question is addressing an extremely important field.
Thank you.
I know we're over time, but I just want to take, I guess, the moderator's prerogative to ask maybe some closing questions. I guess one is at this difficult interface of the direct-to-consumer space, whether it's probiotics or devices that modulate the vagus nerve, through minimally invasive approaches. I'm just interested in the thoughts that each of you have, about the challenges, of how we deal with, these technologies that are now coming to consumers, with minimal levels of regulation, level 1 evidence and oversight, and the potential for benefits, but also the potential for real harms. Kevin, I'm wondering if you've thought about this. Obviously, you're very far along, in terms of moving these technologies out into the public.
Yeah, I think about it every day, and I don't have great answers for you. You have patients in many cases who are facing bankruptcy from trying to go down traditional paths of using biologics to treat their autoimmune diseases. All of these agents, as I mentioned, have black box warnings. They work in 40% of the patients. And then patients read they either read the basic science that's known, or they read about the fact that TENS units are FDA approved for other conditions. And, can I try it? Now, it's a difficult, can I take a probiotic? I can buy, should I eat more Greek yogurt? We are at a time, and I think your question is framing what you said in your opening remarks, that our cultural changes are racing ahead of our evolution, and our technology is racing ahead of our cultural changes. So the question is, I don't have a good answer on how to confront that. All I can do with my colleagues is go as fast as we can in the lab to try to nail down mechanisms that are informative to these answers. We don't have them all.
Do these discussions come up at Altos or in other forums?
Well, you cannot expect to have an answer if Kevin doesn't have an answer. Right? Families are desperate. They have kids that they show behavioral abnormalities, and they are willing to try anything and everything. The way that I see it is some of our discoveries started in our case in 2016. And 6 years later, there's 2 studies in humans already. I think if we are just grasping at what the medicine could be, I think we need to have better mechanisms. Maybe the medicine will not be a microbe. Maybe it will be a metabolite that the microbe produce. If I understand the circuit, maybe I can use, bioelectronics, neuronal dust, to specifically stimulate in the gut or in the brain the circuit that is driving this. But the kids, the families are desperate. Some of the probiotics that people use, microbes are dead. You don't even know what you get there. Incidentally, I receive emails from people that they say that they actually feel better and they improve their behavior. But this has to be done in very controlled trials. And many of us in the field, not only we are facing this problem with the society, some of us, or the field, the microbiome field, is also facing the problem with the neuroscience community, that some of them still cannot conceive that a microbe in the gut or a metabolite produced by a microbe could be leveraged to modulate the brain.
Yeah, Kevin?
This is a huge problem that is not being driven with a financial profit motive. You're talking about probiotics that are generally available. You're talking about over-the-counter 10 units that you can buy on Amazon for $20. So to figure this out, to get the answer to your question in an ethical, scientifically rigorous way, it's going to have to be driven by governments or academics or the private sector.
Yeah, I do think it highlights sort of this the question of, do we have an appropriate intersectoral framework, a regulatory environment? Because as you pointed out, this technology is racing ahead. It's moving towards consumers. We can take a very passive approach and hope that it works out. But, that runs significant societal risks and harm at many different levels. And so I think that's a gap analysis that we have for many different technologies. Just a final question to Sriram, since we seem to let you out of that last discussion, is it's really fascinating to think about the potential for machine learning and AI to really take that holistic view. That's very difficult to do in highly specialized fields, and think about the challenges, not just at the individual cellular level, the organ, the individual host level, and then at a population level. What are at a very high level, what do you see as the biggest challenges of moving that forward? Is it data? Is it sort of the computational frameworks? Or is it something else?
Yeah, that's a great question. I think one challenge is, I think, quantifying all the things that, as humans consume or do, like, for a mouse or cell lines, we can say what's in the media or what's given the feed. For humans, it's really hard to quantify all the things that they do, like the environment they're in, food they consume. So I guess you can imagine maybe an app that notes down the person's diet while they're on a clinical trial or, taking certain medications, and then we could mine all those data together. So I think it's easy to measure a person's genetics right now, but it's hard to measure all the exposome or the environmental aspects. So I guess if you could quantify that too, I think that's the missing piece. We could then put them together, to make better sense of all these complex interactions between environment and diet. And I guess for the last one, I did have a quick comment in that you mentioned about all these new emerging technologies that people are using. I guess a good thing about microbiome or metabolic supplements is that sort of non-toxic. When people want to try it, it's fine. It's not going to do any harm. They're taking a vitamin supplement or a harmless bacteria. Then I guess but
Yeah, well, I'm not sure they're completely.
That's true.
Not
They do
toxic.
have. Yeah.
So I think that we've certainly seen potential risks from what were assumed to be benign nutritional supplements.
That's true. Yeah, I agree.
But in any case, I know I said that was going to be the last question, but I'll make this the last question. I'll take this prerogative. And I know, again, at 1 o'clock, we need to take a 30-minute break before this next session. Yeah, I have 2 children who were born profoundly deaf, and our family are the beneficiary of cochlear implants. And Graham Clark and others spent a considerable period of time decoding the frequencies and the coding algorithms that were required to process electrical stimulation of the cochlea into definable auditory signals that could be understood as speech. With in the context of devices and the vagus and the promise of the vagus to be able to really sort of not have a blunt approach, but a more specific and targeted approach, do you think that there is that potential, or much as we've seen with cochlear implant, or is it just too complex, too geographically scattered over a broad space of the body to allow us to assume that we can take advantage of what we understand about the vagus through the perspective of the device? Or are we ultimately going to require better pharmacotherapeutics?
Yes, it's possible. I'm confident it's possible for a number of reasons. The first is, even in the example of the cochlear implant, what's so striking about that neuromodulating strategy is how few neurons and how few channels you actually need to get usable speech interpretation. Why is that? David Eagleman, I think, says it best. The brain is a perfect pattern finder. It finds the patterns. So what we have to do through sensory substitution, as it's called, and of course, again, these are FDA-approved devices that restore hearing to the deaf and sight to the blind. And right around the corner will be devices that restore movement to paralyzed patients. It's been done already with spinal cord stimulation and with direct brain-computer interfaces, brain implants. It works. And the criticism of some in the news recently for racing ahead without knowing all the neuroscience mechanisms, there's a counter argument to that. First of all, it already works. You can go online and find plenty of videos of patients who can see through their tongue or through other parts of their body receiving the sensory input from the TV camera. You can go online and see patients who were paraplegic are now walking because they are receiving input through their spinal cord. And you can go online and see quadriplegics operating wheelchairs or pouring a cup of coffee or a cup of marbles. So it works. We don't know all the mechanisms. I think the answer to your question is we will have devices that make these things work before we completely understand all the mechanisms, because the brain is a perfect pattern finder and the brain will do the decoding.
Well, with that, I want to thank everybody for fabulous presentations. All right, we will reconvene at 1:30. 1:40.
All right, good afternoon, everyone, and welcome back. I think by our agenda times, we can already tell that there's a lot of really great and exciting discussions and conversations happening for our topic today. My name is Andrew Bremer. I'm a member of the staff team here at the National Academies. I'm pleased to welcome you back. I just want to make a few quick notes about some housekeeping items before we transition right to the next session. We definitely want to continue and encourage the active discussion and questions. For those of you tuning into the live stream, you can place your questions for today as well as tomorrow into the Slido that's directly beneath the live stream. For those of you on Zoom, please feel free to use the raise hand function. We have folks that are keeping track of those so that you can be called upon during our question and answer session to answer your questions. And then, of course, for those of you in the room here, we have the microphones that you can step up to once the Q&A session begins. I also want to note that the workshop presentations and discussions are being recorded and will be made available after the workshop. The discussions will also be recorded in a workshop proceedings in brief, which will be published. It's slated to be published early next year in the early spring. With that, I'm pleased to turn it over to Dr. Diane Delis, who is going to be moderating our next session. Take it away.
Okay, great. Thanks. And welcome back, everybody. I think our first session was super exciting. It will be hard to top that, but I know that we have two amazing experts coming up in this next section to talk about the gut-brain axis in more detail. And so just as a reminder, I'm Diane Delis. I'm from National Defense University, and I'll be chairing and moderating this session, hopefully with some help from the other members of the committee. So our first speaker is Dr. Elaine Chow, who is the DeLoghi Associate Professor of Biological Sciences, and she leads a laboratory studying fundamental interactions between the microbiome, brain, and behavior, and their applications to neurological disorders. Her laboratory is mining the microbiota for microbial modulators of host neuroactive molecules and investigating their impact on the immune system. Dr. Chow has a long list of awards beginning from her very entrance into neurobiology and receiving her PhD, which you can read in the materials. And so without further ado, I'm going to turn it over to her to start with her slides. Dr. Chow.
Great. Hello, everyone. Thank you very much to Dr. Long and the organizers for this opportunity to share with you some of our research interests. I'm really excited to be able to tell you a bit about what we've been up to. As you've heard already, there's a lot of interest in microbiome interactions with the nervous system as a way to intervene and influence the brain and the behavior. My lab and others are really interested in these fundamental questions of what are the effects of normal microbes or indigenous microbes on the development of the nervous system? How do they actively communicate with the nervous system? How do microbe nervous system interactions interface with brain and behavioral networks? And how do microbe nervous system interactions impact health and disease? And when we can, we'd like to also consider why it is that microbes seem to be able to elicit such striking effects on the brain and behavior. Is it really just, a side effect of their microbial metabolism, or is there any benefit at all for them? Some of the themes that we and others are pursuing are the notion that microbes can influence neuroimmune and neuronal development during early life. That aside from developmental processes, microbes can also produce and regulate biochemicals that impact active signaling. That we hope to be able to use our physiological metadata from really defined microbiome systems to inform in silico models for the gut-brain axis. And we also study the microbiome at the interface of gene-environment interactions, especially in the context of a neurological disease. And we have a bit of evidence today that microbial modulation of neurochemicals can impact their own community membership, hinting at perhaps a beneficial role for this function. And so what I'd like to do in this short time is just to share with you a few vignettes on a couple of these themes. And I'll start with this idea of active signaling between microbially produced or modulated molecules and the nervous system. And so to start out, one of the first questions we asked was really with whether microbes indigenous to the gut microbiota could modulate neurotransmitters. We sought this out by studying germ-free models compared to mice that were conventionally colonized. And consistent with existing metabolomic data, we saw that germ-free animals had much lower serotonin levels, a popular neurotransmitter that's highly produced in the gut and also elicits important functions in the brain. We saw that these decreases, microbiome-dependent decreases or increases in the case of colonization, were localized to the colon. And we started colonizing with select microbes or communities of interest. And we're lucky to find a particular community that we call spore formers here, donated by Kathy Nagler, that restored serotonin levels back to controls. And so with this work, we delved in more deeply to find how this group of microbes was modulating serotonin. And in short, we found that they were influencing the synthesis of serotonin from the gut, in particular these endocrine cells in the gut called enterochromaffin cells. And so to summarize this work, which was published now several years ago, found that certain groups of bacteria that are normal to the gut microbiome, both in humans and in mice, produce metabolites that signal to these gut endocrine cells and cause an increase in serotonin production. Serotonin is released toward the tissues, so the host side, and performs its usual functions, including stimulating enteric neurons and promoting hemostasis of platelets. And in so doing, we found that we could use the microbiome to control these physiological functions in a serotonin-dependent manner. And so this is just one initial work from the lab, really inspired by many others that preceded us, including Dr. Mark Light, who I saw will be speaking tomorrow. But it really motivates our interest in studying how microbes can regulate neurochemicals more broadly. In addition to serotonin, there are several others which microbes are able to regulate. So from there, we also examined why, the question of why do bacteria modulate so much of host serotonin? And in our hands and in others, it was up to 60% of serotonin levels were dependent on the microbiome. And we were inspired by these early studies showing that not only is serotonin released back into our own tissues in the bloodstream, but some serotonin is also co-released back into the lumen of the intestine where the microbes reside. And so work by Dr. Jonathan Lynch and Thomas Fung, who are former postdocs in the lab, really sought out to study whether microbes can regulate serotonin because they're able to use it directly. And so not only did the microbiome control host levels of serotonin, stool content of serotonin was also regulated by the microbiome. And so in work from a few years back, we either supplemented with serotonin or raised genetically engineered mice that had elevated gut levels of serotonin to ask, how does the microbiome change if we elevate serotonin levels? And after sequencing the microbiome, we found that in all cases, whenever we increase serotonin levels, we enriched for the particular microbes, the spore former community that we previously found induces serotonin. So this was just a bit of a hint suggesting that microbes that induce serotonin benefit when serotonin is around. We then searched all available microbial genomes for anything that looked like a serotonin response element, like a transporter or receptor. And hopefully not coincidentally, we got one hit, and it turned out to be the most prominent member of the spore-forming community, Tracy here in pink. Which had this uncharacterized protein that looks really similar to the mammalian serotonin transporter on the left. And so in following experiments, we grew up Bacter, and we showed that indeed it can import serotonin directly. It can consume it in culture. It can uptake radio-labeled serotonin, a fluorescent analog of serotonin. And if we took the sequence of the protein and put it into another microbe that typically doesn't interact with serotonin, we saw that it conferred the ability for this other microbe to now import serotonin as well. And so this was really exciting in suggesting that microbes that induce serotonin, at least some of them, may actually interact directly, may actually do so because they interact directly with it. As a side of this, this protein here, serotonin transporter, is the main target of antidepressant drugs. And so we found that all of these effects could be blocked by SSRIs, selective serotonin reuptake inhibitors. In particular, we used the antidepressant Prozac or fluoxetine. And so overall, we found that this particular bacterium that is part of the community that induces serotonin can actually interact and import it directly. And after importing it, it actually had a beneficial effect in enabling it to colonize the gut much better. So this was all based on our very, reductionist work in the lab in culture tubes. And we are really excited that around the same time, work by Jackson and colleagues was really just finding how drug use of prescription medications was associated with differences in the microbiome. And their line here for SSRIs reported one main hit, which was also Bacter in a set of about 3,000 twins, where SSRI use was linked with low levels of Bacter. And this is what we saw as well using this reductionist approach. So we're really excited about this opportunity to study how changes in the microbiome, specifically in Bacter interactions with antidepressants, can influence their efficacy or their side effects. And so this contributes to a larger goal to understand how microbial interactions with neurochemicals and drugs that target neurochemicals can influence disease and health. So from there, we started with this idea of studying neurochemicals with the intention of understanding whether they could influence neuronal activity as a way for the gut microbiome to influence the nervous system. And so slowly but surely we're moving in that direction. And we have here some work ongoing on vagal neurons, which extend directly into the GI tract and innervate the brainstem. And so work by Kelly Jamieson in the lab is finding that if microbiomes are depleted, activity of vagal neurons is substantially decreased. She can also see this when she perfuses the gut with antibiotics to deplete microbes. We see that with perfusion of antibiotics, vagal activity decreases, and vagal activity can be re-stimulated by reintroducing filtered metabolites from the microbiome here in the green lines. And so this means that there are chemicals in the gut that are regulated by the microbiome and influence gut to brain signaling via vagal neurons. And we're currently dissecting the metabolites responsible. And so this contributes to a broader goal to identify the specific microbes, microbial products, and vagal or dorsal root neuronal subtypes that mediate microbiome gut-brain signaling. And so I've told you a little bit so far of just this interest in active signaling between microbes and the nervous system, kind of hijacking typical chemosensory signaling from the gut to the brain. And I'd like to just briefly, a comment that aside from active signaling, the microbiome can also elicit important effects during early development. And so for the interest of this group, I'm not going to share the work in this area, but just to say that this even begins prenatally. So some of our recent findings are that the maternal microbiome during pregnancy regulates metabolites that can enter the fetus and access the fetal brain directly to influence axon outgrowth from there. And so moving on, aside from studying typical interactions between the microbiome and nervous system, we also try to understand how we can leverage these understandings to impact health and disease. And so one example of a study that we've pursued so far is really leveraging a reverse translational approach, an intervention called the ketogenic diet, which has been used for over 100 years to treat epilepsy. So about 1 in 3 people with epilepsy do not respond to existing anti-epileptic drugs. And in these people, especially in children, they're placed on the ketogenic diet through the healthcare system. So it's a rare case where a diet is used as a hardline treatment rather just than, a recommended lifestyle change. Response rates vary, but can be quite good for some people. However, patient retention is really low because of how severe it is. So oftentimes they're started at a 4-to-1 ratio of fat to everything else. And you can imagine how difficult this is, especially for children, where carbohydrates are probably everything they love in pasta and bread and cookies. And so we have some work starting with animal models to show that the ketogenic diet changes the microbiome, and those microbiome changes are necessary for conferring seizure protection. So on the left here is one case where we're measuring the intensity of current that's required to elicit a seizure. And then on the right was a genetic model for a very severe form of epilepsy. And in both cases, we applied the notobiotic approach of colonizing with particular microbes of interest and found a pair that restores seizure resistance. From there, we again used a metabolomic approach and sequencing approach to identify particular species and molecules from the microbiome that predict seizure protection with over 94% accuracy. And based on these findings, we could then use either microbial approaches such as transplantation or probiotic treatment, or pharmacological treatments based on the metabolomic data that we received to design interventions that mimic the anti-seizure effects of the keto diet, even in animals that are on a typical, like a control diet. And so this was a bit of evidence that we could use the microbiome to either enhance the keto diet and make it more efficacious, or even, pie in the sky, potentially replace the keto diet in terms of being able to confer seizure protection. And so we have ongoing studies with Dr. Matsumoto and the keto diet program at Mattel Children's Hospital at UCLA, as well as a few other hospital partners on studying how this translates to the human condition. But overall, this contributes to our goal to understand how microbes can impact behaviors that are relevant to neurological diseases. So from that, we're really just excited about the prospect of understanding, more details about inter-organ communication, and in a way that could potentially lead to new approaches for treating symptoms of disease. So with that, thank you very much to lab members who I've tried to highlight along the way. Christine Olson, Thomas Fung, Helen Vong, and Jessica Yano led the work that I described, and also to our funding support. I'd be happy to take any questions.
Elaine, thank you so much. And I know there's going to be a lot of questions because I've written down a few myself. If you could just hang in there, we have one more speaker and then we'll do the joint questions at the end, if that's okay.
Sure, thank you.
Okay, perfect. Thank you. So our next speaker, Sarkis Masmanian, is the Lewis and Nellie Sue Professor of Microbiology in the Division of Biology and Biological Engineering at Caltech. He received his doctoral training in microbiology and immunology and was the Helen Hay Whitney postdoctoral fellow at Harvard Medical School. His lab studies the human gut microbiome with a focus on developing novel therapeutics for such things as autism spectrum disorder. We talked a little bit about that this morning, and Parkinson's disease. So with no further ado, I will turn over the mic and for our second speaker. Thank you.
Thank you, Diane, and to the organizers for allowing us to present our work. It's a really exciting forum to discuss the possibilities of the microbiome and its future on potentially improving the lives of individuals with neurological conditions. So I'm hopeful everyone can hear me, and I also hope that you can see my slide. You can see my cursor. Is that visible to you on your end?
Yes, we can hear you very well, and I can see your cursor in your slides.
Terrific, thank you. And so our lab works on a number of different areas involving the gut-brain connection. Today, I'll talk to you about our work in Parkinson's disease. And so, as you know, Parkinson's disease is a movement disorder. It's characterized by changes in various different aspects of motor performance, including tremors and gaits, but also leads to posture instability and other issues. That's how it's diagnosed and characterized, but it's very, very well known that Parkinson's is comorbid with many non-motor symptoms that are listed here, constipation being the one that really drew our attention to this area. And so we had started working on autism spectrum disorder in animal models and got bold, if you will, and many years ago decided to test the hypothesis that maybe the microbiome, or at least the gut, was involved in animal models of Parkinson's disease. And it's really for this reason that upwards of 80% of individuals with Parkinson's will have constipation, and in some cases, clinically diagnosed constipation. And what's quite interesting is that oftentimes the constipation precedes the motor symptoms by years, if not decades. There are other prodromal symptoms as well, such as REM behavioral sleep disorder and olfactory deficits. But again, this very curious notion that there is GI, gastrointestinal involvement in Parkinson's at least suggested the possibility at the time that the microbiome or gut bacteria were involved. And this notion that the gut is somehow associated with movement disorders is nothing new. There's an essay that's over 200 years old now from James Parkinson, where he talked about a number of different things, including essentially case studies of individuals that he was following that he called shaking palsy, and then essentially identified that many of them had what would be constipation, and essentially and other forms of GI disturbances. And at least in one individual, treated them with essentially what is a bowel cleanse or a stool softener, and then that led to improvements in the hallmark symptoms of shaking palsy or that movement disorder. And so again, this is something that neurologists and certainly patients and caregivers and families have known about for quite some time. It's really in the past, I'd say, 10 to 15 years that there's been more and more of a highlight on the potential non-brain and non-motor symptoms of Parkinson's. And if Parkinson's were to originate in the gut, I think there's a number of different ways that the intestines can send their signals to the brain. We've discussed some of these already in the workshop, but it could be through inflammation, it could be through the vagus nerve, or potentially other mediators, such as direct microbial signaling to the brain. But this notion, again, that the intestines may be involved in Parkinson's was identified, again, maybe about 20 years ago by Heiko Brake, who hypothesized that potentially because of the gastrointestinal symptoms, and because the fact that those symptoms oftentimes precede motor symptoms, that maybe the origination of some forms of Parkinson's may be in the gut. And this was corroborated in work that he did where he identified the classical pathology of Parkinson's, which are Lewy bodies or aggregates of alpha-synuclein. I'll talk about that in a minute. He identified it in the gastrointestinal tract post-mortem of individuals who had Parkinson's, and then proposed what is Brake's hypothesis, which essentially suggests that maybe there's a non-brain origin that involves this hallmark pathology and the symptoms associated with the pathology that may begin in the gut, work its way up into the brainstem, into the brain, and then spread throughout the brain. And this is actually the brain spread portion of which are staging criteria that are used in the clinic. And so there's now a lot of evidence in humans, the associative evidence in humans suggesting that indeed there may be spread of pathology from the gut to the brain. There's interesting parallels between individuals with Crohn's disease and Parkinson's disease. There's also epidemiologic data that individuals who are vagotomized can be protected from Parkinson's. And in animal models, mainly rodent mice and rat models, seeding the gut with this pathology, with alpha-synuclein, results in spread to the brain and motor symptoms, which can be mitigated by vagotomy in animals. And so again, I don't think anything has yet been proven, but certainly there's a lot of evidence now suggesting that potentially some forms, as I mentioned, of Parkinson's may begin in the gut. And this doesn't exclude other non-brain origins sites, such as the olfactory bulb or even the skin. And so, again, it's widely believed that Parkinson's is promoted, if not even caused, by aggregates of alpha-synuclein, it's a neuronal protein that if it misfolds and is not cleared by cellular machinery, such as the proteasome or autophagy, that these fibrils then aggregate and essentially cause neuronal distress. There are certain neuronal populations which appear to be more vulnerable to alpha-synuclein aggregates, namely dopaminergic neurons, and they seem to be affected in particular parts of the brain that are at least associated with the motor symptoms, and that's the substantia nigra. But certainly there are other neuronal systems which are not as well characterized, which are involved in Parkinson's and other brain regions where alpha-synuclein aggregates may play their role. And so many of the features of human Parkinson's can be modeled in mice that overexpress wild-type human alpha-synuclein. So just making more of alpha-synuclein leads to its aggregation, essentially overwhelming the cellular machinery that would clear these types of aggregates. And we used a mouse model that was developed by Eliza Messelet many years ago, which showed not just the motor symptoms, but also gastrointestinal symptoms. So we figured this would be a good place to test our hypothesis that the microbiome is involved. So again, these are alpha-synuclein overexpressing mice that express alpha-synuclein in all neurons of the body. And these mice have motor deficits. And so again, we're just reproducing data other people had shown that the alpha-synuclein overexpressing transgenic animal has a motor defect. In this particular case, a beam traversal test where we place a mouse on a beam that gets progressively narrower, measure the time it takes to cross the beam. These animals will take longer to cross the beam than their wild-type counterparts. SPF stands for specific pathogen-free. These are mice that have a standard laboratory microbiota. So we obtained these animals, we re-derived them under germ-free conditions, essentially keeping genetics identical, just removing their microbiomes. And you can see that the alpha-synuclein overexpressing mice that do not have a microbiota that are germ-free cross the beam at what looks like a normal level. We use multiple different behavioral tests. This is the pole descent test, where we take an animal, put it on top of a pole, and measure the time it takes to grasp and descend that pole. Alpha-synuclein overexpressing mice have a defect if they have a complex microbiota, but not if they are germ-free. And then the test of fine motor skills. Here's an adhesive removal test. We take an adhesive, put it on the nasal bridge of the animal. And again, the transgenic animals that have microbiota take longer to grasp and remove that adhesive compared to the germ-free ASO animals. I'm not showing the data here for the sake of time, but gastrointestinal transit as well as fecal output are also affected by the microbiota. And also water content is lower in the fecal pellets of mice that have alpha-synuclein overexpression if they have an SPF microbiota. Essentially, this is modeling constipation in these mice. And so we want to also look at the hallmark pathology, which I mentioned is this the aggregation of alpha-synuclein. And so using an antibody that recognizes the aggregated form, so there's a conformation-specific antibody that labels in red, you can see widespread aggregation of alpha-synuclein, this is the caudate putamen of mice, but the same animals under germ-free conditions do not show these aggregates of alpha-synuclein in the brain, and it's also true for the gut as well. Again, I'm not showing that data here. And what's really interesting is we wanted to essentially do the sanity check and know that whether germ-free rederivation didn't change levels of expression of the transgene, and that's indeed the case at the RNA level, also at the protein level. By Western blot, you can see the soluble alpha-synuclein is identical between germ-free and SPF mice, quantitated here. But these aggregates are insoluble, they're protease resistant, and so when we look at what portion of the alpha-synuclein that's produced aggregates, there's more aggregates in the SPF animals than the germ-free animals, consistent with the motor data. And so we used antibiotics and showed the same effect. We can recolonize germ-free animals and restore the motor deficits. We can even transplant microbiota from humans, both the Parkinson's and healthy controls, into the animal models. And if mice receive microbiota from a Parkinson's patient, their motor symptoms are worse than mice that receive microbiota from a healthy control. And so in addition to the microbiota experiments, over the years, we've identified bacteria which can promote symptoms of Parkinson's, and we have a good idea of what those molecular mechanisms are, and we continue to work on that, and that's published. I won't talk about that here. We have unpublished data, early evidence that there are anti-inflammatory organisms which can prevent the symptoms of Parkinson's and the neuroinflammation that's associated with that. But what I want to talk about today is a recently published study where we wanted to use a dietary intervention and ask whether or not it changed neuroinflammatory parameters and also improved symptoms as can be modeled in mice. And so a lot of evidence has shown that short-chain fatty acids, which are produced by gut bacteria from dietary fiber, have pretty robust effects on the immune system, particularly the gastrointestinal or mucosal immune system. The major short-chain fatty acids that are produced from dietary fiber are acetate, propionate, and butyrate. And it appears that these levels are reduced in individuals with Parkinson's disease. There's also evidence that the microbiota from Parkinson's patients in bioreactors metabolizes fiber less efficiently than microbiota from healthy controls. There are epidemiologic data suggesting that a diet, Mediterranean diet or a high fiber diet correlates with lower incidence of Parkinson's and improved cognition. And previous work, both from our lab and from other laboratories, show that short-chain fatty acids, again, produced from dietary fiber, can affect microglial activation. I'll talk about microglia in a minute. And so what we wanted to essentially model is whether or not a dietary intervention with a prebiotic or high fiber diet, which elevates short-chain fatty acid levels, leads to improvements in motor symptoms via modulation of inflammation in the brain. And so we work with Bruce Hamaker at Purdue. He's an expert in food omics, and he engineered a diet specifically for our purpose. And the criteria that we asked him to evaluate were, is there a mixture of fibers which increases short-chain fatty acid production via increasing the levels of short-chain fatty acid producing bacteria. And again, for the interest of time, I won't go through this, but it was an iterative process where he and his team formulated various diets. They sent it to us, we put it into mice. He used bioreactor studies to initially screen them, and then we identified a particular diet which indeed did increase short-chain fatty acid levels in the intestines, and you can see those here, and you can see the difference. There's no difference between wild type and ASO, which is what we were sort of going for, except for this level of propionate. The goal here again was to identify a mechanism to increase these short-chain fatty acid levels, and then see how that affected an ASO-specific outcome. And so this isn't projecting well, but I can essentially tell you that as predicted by the bioreactor studies and then validated here in mice, that the prebiotic high fiber diet did change the microbiome, and it changed it in a way where it did increase the levels of short-chain fatty acid producing bacteria. And what is missing here, this just didn't transfer well in the slides, what's missing here is a principal coord analysis where the dietary effect was much stronger than the genotype effect. So the microbiome is different between wild type and ASO mice on a control diet. When we used the prebiotic diet, both microbiomes shifted and the magnitude difference between them was increased by the diet. So the diet effect is actually much stronger than the genotype effect in this mouse model. And then, of course, we went to the motor tests, and indeed, the prebiotic diet did improve most of the motor symptoms found in this mouse model. So this is the data I've already shown you for pole descent. You can see the ASO animals take longer to descend the pole. If they're on a control diet, if they're on a prebiotic diet, you can see that the ASO animals can descend the pole, again, similar to wild type animals. We can look at the time it takes the mice to cross the beam. This is the data, again, similar to what I've shown you before, and that phenotype has improved. We look at a number of different parameters. We can look at the number of steps the mice take to cross the beam, or even the errors that they make per step. And again, we see a phenotype there, and then that's restored. And then we see a trending improvement in adhesive removal on the animals that were given the prebiotic diet. When we look in the substantia nigra of these animals, here we're using dot plot to identify alpha-synuclein aggregates. You can see, again, these aggregates been shown by many other groups to be found in this animal model in this particular brain region. Much fewer aggregates, in fact, similar to wild type levels in animals that were given the prebiotic diet. And so a lot of work from the Alzheimer's field in particular, but also from mouse models of Parkinson's, have shown that microglia, or brain resident innate immune cells, seem to be quite important in modulating both the neuronal defects that are found in these animals, but also the behavioral defects, whether it's cognition, Alzheimer's, or motor symptoms in Parkinson's models. And so we wanted to look at the effects of this prebiotic diet on the functionality of microglia, also because of the fact that it had been previously shown short-chain fatty acids modulate their activation states. And so just very briefly and at a very high level, microglia can take either a pro-inflammatory or an anti-inflammatory profile. These are really crude distinctions of what microglia can do. In fact, they're, in addition to being immune mediators which help clear viral or bacterial infections, they have a lot of other functions in the brain that were highlighted in this very recent review article, or essentially an opinion by a broad number of investigators who research microglia, and trying to understand what are all the different roles that microglia play. And in fact, they're quite important for neuronal health and for neuronal branching and how neurons actually talk to each other. And so, I think there's a lot of work going on outside of Parkinson's, outside of the microbiome, and trying to understand how relevant these cells are to various different activities in the brain and potentially the behaviors that are associated with those activities. And so with that principle, we looked at microglial activation. Here we're looking at the substantia nigra and the striatum. And when microglia become activated, their cell body diameters increase. You can see these are actual individual microglia. These are live cells that we imaged using three-dimensional reconstruction. And looking at their cell bodies, again, the activation results in the ASO transgenic background on mice with given a controlled diet. But the mice given the prebiotic diet, their microglia appear to be much less activated via this imaging strategy. And what's also known is that when microglia do become activated, they branch less, and the number of segments and branch points decrease. And you can see that all in the control fed mice, but the prebiotic fed mice, their microglial parameters look like they're anti-inflammatory. Again, there's data points missing here that just didn't transfer well in the slides, but clearly there's no defect between wild type and ASO mice on a prebiotic diet. And so we wanted to get a little bit more granular and try to understand really what the microglia are doing. And so we performed a single cell RNA-seq experiment where we extracted the microglia and then profiled them on an individual, profiled their transcriptomes on an individual cell basis. The first comparison I'll show you are wild type and ASO mice on a controlled diet. So what it had, the microglia from this mouse model had previously not been profiled by single cell RNA-seq. And what we see, or what one would expect, right, is that there are a number of pro-inflammatory markers that are elevated in mice that overexpress alpha-synuclein compared to their wild-type counterparts. And then when we do gene enrichment analysis, you can see that there are 163 different genes that are found in various different pathways that are listed here, which are upregulated in the ASO mice relative to the wild-type animals. The next comparison is the ASO animals either fed the control diet or the prebiotic diet. So again, both of the groups have the alpha-synuclein transgene, a number of differentially expressed genes. But here what we see are that the prebiotic diet is down-regulating inflammation on a gene level. And then when we look across multiple different pathways that are listed here, you can see that the prebiotic diet is really anti-inflammatory and down-regulates multiple different inflammatory or stress pathways. And what's also really interesting is that the phenotype of these microglia, which are expanding on the prebiotic diet, appear to be disease-associated macrophages. And DAMs are a subset of microglia, which have been shown again largely in the Alzheimer's literature, which have been shown to be protective against cognitive impairments. And so the prebiotic diet is enriching for this population of microglia, this DAM population. And that may be leading to these benefits in the motor performance that I showed you. So we don't know if it's the DAM or some other population of microglia, but ultimately we want to understand, are microglia required for the protective effects of the prebiotic diet? So we use the compound PLX5622, which depletes microglia, so we can essentially knock down or reduce the levels of or the numbers of microglia in the brain, and then see what the prebiotic diet does. And so in the substantia nigra, we get about a 60% depletion of microglia. And that leads to, that in and of itself leads to less alpha-synuclein aggregation between mice on the control diet and mice on the control diet that were just given the drug to deplete their microglia. But really what's interesting is, again, that microglia are required for the beneficial effects of the prebiotic diet. So in the pole descent test, you can see, again, this is data I've already shown you, is that the mice have a motor performance phenotype. If we deplete their microglia, again, for mice that are on a control diet, the mice still have motor deficits, meaning that at this age, at least, which is about 5 months of age, these mice do not, their motor defects are not driven by their microglia, because again, microglia are gone, but the mice still have a motor deficit. Again, this is data I already showed you, is that the prebiotic diet is beneficial and reduces the motor symptoms. But if we remove the microglia on the mice that were given the prebiotic diet, now their motor symptoms reemerge. And that's the same for the time to cross the beam. Again, the prebiotic diet is beneficial, but in the absence of microglia, the prebiotic diet is no longer protective. And this data, again, suggests that microglia are a target of the high fiber diet, or at least are required for the beneficial effects of the high fiber diet. And subsequent studies will try to understand what are those specific pathways and how do the signals from diet reach the brain and impact microglial biology. And so I'll just end with just this notion that, again, Parkinson's disease is associated not just with GI symptoms, but with really characteristic changes in the microbiomes, a dysbiosis that appears to be not entirely, but largely replicated across a number of different studies. And so that's heartening that there may be a particular signature of Parkinson's, which is, pretty unique. As Mauro mentioned, if you look across many studies in autism and a number of other disorders, you don't see much concordance in the literature for organisms that may be depleted or enriched. But in Parkinson's, like, same organisms keep coming up cohort after cohort. So there may be some information there that we can leverage for future studies. But again, in this study, what we were interested in is how do you, what are the beneficial effects of a high fiber diet? And indeed, what we showed is that there are improvements in motor function, likely working through microglia. And so I'll just end by thanking Rima Del Haq, who's a recent graduate from our lab. She received a PhD, and she drove all the dietary studies that I showed you today. And Tim Sampson, who's a professor now at Emory, started this research when he was a postdoc in our lab, and he was the first to discover that the microbiome is important in the mouse model of Parkinson's, which again has been reproduced by a number of different labs as well. So Anna, Joe, Olivia, Taryn, and John are members of our lab who've helped along the way. And we have terrific collaborators who provide invaluable help. Couldn't have done the research without them and our funding sources. Thank you.
Wow, that's a tour de force by both speakers. Thank you so much. Fascinating data. I know there are questions, so we're going to start here in the room first, and then we'll go online. You can either make your way to the microphone or come up and sit with me. Either one. Okay, so Jason.
Hi, fantastic talks. I learned so much that I'm sure I'm going to be thinking about this for a very long time. But I'm going to start off with just a couple of very naive questions, or one in particular, which is, so alpha-synuclein, obviously a strong correlate with microbiome. Have we seen other types of correlates with like tau or amyloid or polyglutamine diseases?
In case they couldn't hear the question, is there a connection
I'm sorry.
similar to the alpha-synuclein connection with tau, amyloid, or polyglutamine
disorders?
diseases?
Yeah, so we've worked but not yet published on mouse models of Alzheimer's, where we've shown that depleting the microbiome leads to less A-beta 42 production in the brain. San Sisodia at the University of Chicago has published on this, where antibiotic treatment leads to improvements in A-beta aggregates. And David Holtzman has shown in models of tauopathy that antibiotic treatment improves behaviors as well. I'm not sure if that data is published. My sense is, at least from the data that I've seen to date, that the effects on alpha-synuclein seem to be more robust than that of at least A-beta, where there's more literature than other amyloid-like diseases. And again, this has been broadly reproduced, not broadly, but it's been reproduced by a number of different laboratories. I'm hopeful that it'll be broadly reproduced over the years. And so it looks like the picture's emerging that maybe there are effects on these aggregate or diseases of amyloids or associated with amyloids that could be broader than the ones that have been more studied. And I'll just also mention, because it relates to Parkinson's, is that bacteria produce amyloids as well. And one of the other studies that I alluded to were that we've identified a bacterial amyloid, which leads to increased aggregation of alpha-synuclein. So that could be even cross-seeding between a bacterial and a mammalian amyloid. And that's relevant because there appears to be cross-seeding between mammalian amyloids. So oftentimes you find synuclein in A-beta plaques and tau in Lewy bodies. And so I think the picture's emerging that there may be sort of this crosstalk between amyloid proteins between bacteria and the mammalian hosts.
Well, as a follow-up, some of your comments that suggest to me that some of these protein misfolding disorders might have a strong microbiome correlate, and that my mind leaps to well, has anyone looked at some of these prion disorders and is seeing if perhaps there are correlates there as well?
So the question is sorry, I didn't catch the question.
Connection between microbiome and prion disorders. Has anyone looked?
In terms of protein misfolding disorders and things of that nature?
Not that I know of. Yeah, not that I know of or come to mind.
And for the other speaker, I was completely first time I've ever heard of anything about ketogenic diets and treatment of behavioral disorders or epilepsy. I was wondering if you're aware of any other examples of that where high protein diets or any of these other sort of what I guess someone would talk about as fad diets, if those have been linked or have been used in the past to treat similar types of pathologies.
Yeah, so the ketogenic diet is probably the most well-established since it's actually used in the healthcare system and is very common, aside from being a fad diet for weight loss. It's actually a hard line treatment for epilepsy. So aside from epilepsy, the keto diet is being increasingly applied to other disorders, including neurodegenerative disorders. But isn't really clinically used for that case yet. I think it's still under exploration. In terms of other diets that are used to treat neurological diseases, I don't think there's one that's so well validated as the keto diet. I think there's kind of anecdotal data with autism that certain restricted diets may be useful for particular groups of kids. And yeah, other data on FODMAPs diet, which is useful for treating visceral pain in IBS, which is also kind of has some neurological underpinnings. And so, yeah, those are a few that come to mind. But I think in starting out in this field, we intentionally chose the keto diet as something to explore, since we already know that it works clinically, but has challenges in its implementation and efficacy.
Great, thank you. Let's go to Elliot in the room.
Elliot Chaikoff, and again, I'd like to thank you both for both outstanding presentations and for agreeing to participate in this workshop on really very short notice. So it was very kind of you, but really fantastic presentations. I have 3 questions that I think could go to both of you. I'm just first curious about whether or not you've observed any gender-specific effects in your studies. A second question goes to looking at your data sets. Clearly the results are significant, but there's a lot of variability in the animal groups. It seems as if there are responders and non-responders, and I'm wondering if you have any insights or thoughts about that. And then lastly, as I look at work in this field on diet and probiotics, I'm just wondering about your thoughts about the kinetics of the effect. You know, in some cases, it seems as if a dietary change can cause very rapid, profound changes in the gut microbiota. And in other cases, it seems as if the effect to restore a dysbiosis through a dietary modification can take quite some time for that effect to take place. And I'm just wondering if you've given that some thought about some of the difficulties or challenges in translating this to clinical studies, again, both for diet and probiotic therapeutics. Thank you again for really spectacular presentations.
Elaine, you wanna start?
Sure,
you
I'll take
go.
I'll take a stab at a few of these. Yeah, so I think the study of sex differences is really important, especially in the context of all of these neurological disorders we've discussed, which do have overt sex biases. Males for autism, males for Parkinson's, and other neurodegenerative diseases, which are more tilted toward females. In the work that we've done so far that I presented with the serotonin and fetal early life work that we've done with fetal brain and also in epilepsy, we didn't see any sex differences. But we have some work that we just recently posted on bioRxiv of a Parkinson's model called LARC2 G2019S, where we did observe sex differences and tried to get at whether the differences were due to hormones or sex chromosomes. And so, yeah, I think on the microbiome side, sex differences in the microbiome have been reported, but I think they're relatively modest compared to other factors, like the dietary interventions or environmental challenges, is my impression. Yeah, and then in terms of kinetics for diet, maybe I'll leave a few for Sarkis to speak to. For kinetics, I think it really depends on the severity of the dietary change and the type of dietary change. For us in the keto diet, which is super severe, it's a very low carb, high fat diet. In mice, it's a 6-to-1 ratio, and in humans, it's 4-to-1. We see that the microbiome shifts within 4 days, and it stays there without further change for months after. I think Peter Turnbaugh had done a study in the human cohort where they were fed really defined plant-based or animal-based diets, where the animal-based one was ketogenic as well. in that case, all the dietary, like the exact meal plan was controlled. And so they also reported changes in humans within 4 days. And so that's just one example, but I know what you mean, where I think it depends on the severity of and type of the perturbation that's involved.
And I'll just, from our end, at least for the Parkinson's model that I showed, the alpha-synuclein overexpressing animal, we have not looked at sex differences because we can't. The transgene is on the X chromosome. And so we use males so that you don't get X inactivation and mosaicism. But we're now using a number of other genetic models which don't have this limitation, and we can look at sex differences in the Parkinson's model. In terms of variability of behavior, especially when it's sort of emotional or even motor behaviors and cognitive behaviors, there's a lot of variability, a lot of spread in the data. I think that's common across laboratories, though at times frustrating. But I think that's an opportunity as well, right? Is because we can potentially think about high and low responders within any one group and then go back and identify, are there microbiome changes which result in, again, within a group, why some animals are performing differently than others. And we started this, we should be better at it actually in our lab, but we started looking at the microbiomes of individual animals within a group to try and see, is that explaining their changes in behavior. In terms of the kinetic effects of diet, or maybe even one step further, what do those effects do to the organism itself? As Elaine said, there are rapid changes depending on the diet. There could even be long-term changes people are now looking at in humans. But as the microbiome changes, we're also obviously interested in how that affects an outcome of interest. And at least my sense is that certain types of behaviors, maybe anxiety, are more tunable in a rapid fashion, because I think that those are more resulting, and a lot of this is speculation. Just to be clear, I think things like anxiety likely result from potentially active signaling between the microbiome and the brain. There aren't really any structural issues in the brain that may be related to anxiety. And so it's more tunable. When you talk about something like neurodegeneration, I think the timelines and some of the data we have suggests is that timelines for interventions, whether it's restorative or even damaging, will take longer because the nature of the pathology just requires more time to either repair or to worsen. And so again, I think it's more context specific in terms of not just what the dietary effects are on the microbiome, but how long does that take, those microbiome effects or changes take to impact the behavior or brain activity.
Question, Elena. You've written, I think, a lot of the publications are getting jumbled up in my mind, but I think you've written about immune imprinting, immunological scarring, or at least those terms have come up. As we've thought about this impact of the microbiota on the immune system, is this, nothing more than the generation of memory T cells, or is it something more than that? These sort of long-term effects of the microbiota, in their interface with the immune system and the immune system's interface with the neurological system.
Sure, I'll take a stab at this. And so I think I can't take credit for the really pioneering findings in this field, but just many groups finding that the microbiome during early life is really important for influencing immune development. And that started with, I think, in the GI tract itself, immune cells that are there, and more recently is extended into immune cells that are resident to the CNS, which Sarkis alluded to, the microglia. So it seems that from work from Sonia Gurel, Prince et al., that they find that the microbiome can influence microglia, and they can also begin to elicit their effects during early development. So we've done a bit of work so far on even prenatal influences of the maternal microbiome, and similarly see some differences in immune cell subtypes, even in the placenta, which is really important for controlling maternal fetal interactions. And so imprinting is also prevalent in that tissue. And so I think there's still a lot of work to be done in the area and how these types of interactions can impact the nervous system in particular.
And if I may just jump in, there's really emerging work, just to add to what Elaine said, there's emerging work now on meningeal lymphatics and how immune cells that don't enter the parenchyma of the brain but get very close within the meninges can send their signals, maybe via even cytokines and other molecules, into the brain that are sensed by neurons. And it's at least been shown that several cytokines, IL-4, interferon gamma, affect social behavior in animals. And these signals may be coming from T cells. But again, it doesn't look like those T cells are in the brain themselves.
Okay, I'm breaking in. Thank you so much for your answers to these questions. And I will say that there are still more questions in the room and online. I'm getting the indication from my National Academy of Science colleagues that we should take a quick break here. I don't know if the speakers can stay on. The roundtable begins at 3 o'clock. And if you are able to stay, there are several more questions. We're going to have a full roundtable discussion, and I hope that you can stay and field some of these additional questions, if that would be okay. Does that sound good to everyone? Okay, thank you so much. Let's all take 10. We'll be right back. And thanks again. Let's give our speakers a great round of applause.